Uh, Good morning to everybody. Today is Friday, the June 19th. This is our last lecture before we dive into the exam. We have a ton of stuff to cover. We need to finish off the rest of our integratory system. Talking about that, if there's time at the end, we have some more histology we can play with. And all of that is about getting us ready for Monday. Again, Monday, we have no lecture. You're going to use the entire class time to take both the lab and the lecture exams. You may take them in either order that you want. Take one first, whichever one you want, you're more comfortable with, take that one first, then take the other one. Take a break in between, however you want to do it. Sleep in, but both exams must be completed within the class time from 8 a.m. to 12:35 p.m. Remember, you must show a picture ID for it to be a valid exam. It can be your student ID. It doesn't have to be your driver's license, although a driver's license is fine as well. And make sure you scan your work area. Again, that's one of those things that can cost you 10% right off the bat. So basically, if you forget to scan your work area, your chances of getting an A, you have to basically ace the exam to do that. So make sure you do those things. Again, maximize.
Quick question.
Yes.
When you're saying scan the work area, I know you mentioned before, like don't scan the walls or the ceiling. So you want just exactly where we're sitting and everything that's like around us.
Well, that's what I care the most about. Yes. I mean, it's okay to, I don't have a problem if you show the room, but I want to see your workspace. I want to see the table in front of you, what's behind the computer. I want to make sure that you don't have a second computer up with information on that, or you don't have a big pad of paper that has a bunch of notes, or your phone sitting right next to you. I want to see that you have a clear work area where you're doing it so that the work that you are presenting on the exam, I know is yours.
Gotcha.
That's what I'm looking for. All right. So again, do that. Like I said, hopefully you won't have any problems. Most students last semester did not have problems. Rarely when the problems come up, if you get kicked out of the exam or something like that, contact Proctorio first. The Proctorio's help is very useful at getting you back into the exam. I do not have the means of getting you back into the exam. I can reset the exam, but I can't get you back into the exam. So you want to talk to Proctorio first if you have any problems, because they will be able to get you back into the exam and do it right away. I want to warn you that the timer doesn't stop. If you get kicked out of the exam, the timer is still going. So you need to get right back in right away so you can continue doing that. And like I said, I think last semester only, I think a total of 4 people got kicked out of it, out of the, 7, 8 exams I had to give between the 2 classes. So it shouldn't be an issue, but I do want to make aware for if it does come up, that is an issue, that is something that you are aware of.
A quick question.
Yes.
How do we contact the Proctorio?
If you get kicked out of the exam, then there is a contact there's a link that you'll click onto to get to Proctorio that they can assist you.
Oh, gotcha.
Okay. So that's it for the exam. I think you guys have seen the practice exam. You know what to expect from that, hopefully, so you'll be prepared for that. The other thing that I want to remind you about, well, not remind you, but make you aware of, is that on Tuesday, we start the skeletal system, and we're going to be learning the bones. And part of that, rather than me just standing here talking about the bones and pointing out this and pointing out this, we're going to try to make it more interactive. So as you've hopefully figured out at this period of time, it is important to be here and be part of the class, be part of the learning process, ask questions and things along those lines. However, on Tuesday, it's going to be even more important to make sure that you are here, because on Tuesday after the lecture, we are actually going to be breaking up into different groups. I'm going to be breaking the class up into different groups. And when we break up into those different groups, breakout rooms, those groups, you're going to be given assigned bones and bone features that you are going to be responsible for labeling on an image, and then you are going to be presenting that starting on Thursday. We will be presenting those, so you guys will be presenting your bones and bone features, so we'll be working together to learn that material. So we'll be forming groups, we'll be doing breakout rooms where you guys will be able to work together during the class time to work on this and make sure you have that. So you'll want to make sure you have, you're here on Tuesday to participate in that, and make sure you have your atlas. If you don't have a photographic atlas yet, you are going to need one, because you're going to need something that is going to be able to show you the bones and show you the bone features, so that as you go through this, you'll be able to label the things and identify the things and know the things. So make sure you have those resources available for you so that you're ready for that on Tuesday. All right? Questions on any of that? All right, excellent. Let's dive in. We left off last time and we were talking about the epidermis. We know that the tissue type is a keratinized stratified squamous epithelial tissue. But as we also talked about, we now know that in that epidermis layer is divided up into 4 or 5 distinct stratas, or really what it should say here is sub-layers. These are sub-layers. So there are 4 or 5 sub-layers of the epidermis, and you are responsible for identifying those histologically, knowing what's going on in those areas, and identifying them. So let's go through those and talk about those. Now remember, as we talked about, we are going to go from superficial to deep because this is, oh, I lost my, there we go, how the skin grows. The skin grows from the deep layers of the epidermis out towards the superficial layer. So it makes more sense to talk about this in the fashion of which they grow. Starting first with our deepest layer, which is known as the stratum basale or the stratum germinativum. Again, as we've talked about, often there are completely appropriate anatomical terms for things that are identical in their meaning, and that is the case here. The stratum basale is the deepest layer. It is the base layer, which is why it's called the stratum basale. It is the stratum germinativum because to germinate means to give birth to, right? You germinate flowers to give life to, and this is where the new cells form. The stratum basale is the deepest single layer of keratinocytes. So what I'm going to do here is I'm going to cheat, grab my highlighter, and if you look, you can clearly, as we've seen, talked about, seeing the edge of the epidermis versus the dermis. That keratinized stratified squamous epithelial tissue is the cell-dense epidermis, and then we have our connective tissues, which are more cell-sparse underneath. Now, unfortunately, the marker of my highlighter is a little thick, so I may be getting a little bit more than one cell in some of these, but you guys will hopefully get the point from what I'm doing here, that that single layer of cells that is there at the base of our epidermis is our stratum basale. These cells are highly mitotic, dividing, these are basically as we've talked about before, are unipotent stem cells. Remember, these are stem cells. They are able to divide rapidly. Wow. Unipotent stem cells that divide rapidly, don't have any other job really. If you think about it, remember we talked about division. Mitosis requires a tremendous amount of energy. And these cells are the cells that are closest to the dermis, closest to the blood supply. Remember, epithelial tissues are avascular. So here is where the blood vessels are located. And so they're getting their oxygen and their nutrients from diffusion. And these are the cells that are right next to it. So they're highly mitotic. They're able to divide rapidly. However, while they're producing millions of new cells every day, every single one of those million new cells is basically a one thing and one thing only, a keratinocyte. And that's all these cells are doing, right? As again, dividing requires a tremendous amount of energy. So all these cells are doing are dividing to produce new cells. All right, so again, these are unipotent stem cells. I want to keep that here, so let's move that over there. That are highly mitotic, dividing rapidly. We'll get to that in a second. And as they divide rapidly, they of course push the other cells that are above them up. So as this occurs, they're going to be pushing the other cells up away towards the surface of the skin as they do that. Now, As we also talked about, while keratinocytes make up about 99% of the cells of the skin, in fact, if we were to throw a dart at the picture of the epidermis, pretty much every cell we see here is a keratinocyte. Remember, we also talked about there are 3 other types of cells in the epidermis as well, and all 3 of these can be found in this area. The Langerhans cells, remember, are also the ones that are called the epidermal dendritic cells. Oops. Those epidermal dendritic cells, they're the ones that the immune cells help to contain histamine, help to be phagocytes, help to provide protection. Our Merkel cells, which remember we also called the tactile cells, those are the ones that give us our fine discrimination. And the melanocytes, which remember are the ones that are responsible for the coloration of the skin. These are the ones that produce the melanin. These also can be found in the deep layers. Now, what I will tell you is that while all 4 of these cell types are found down here in the stratum germinativum, from a histological standpoint, it is possible to identify a Langerhans cell. It is possible to identify a Merkel cell, but I don't expect you as introductory A&P students to be able to recognize them histologically. However, as we'll see, melanocytes are easily identified in the deep layers of the skin, and so that is something you will be responsible for. Let's take a look at this. I need to clear my pretty pictures here for a second, and we can take a look at a different skin. This happens to be thick skin. Again, this is going to have 5 layers here as we look at this, whereas this picture here is thin skin. Notice we can tell that by the top layer, the keratinized cells are much, much smaller. But again, notice most of the cells that we look at here are keratinocytes. So on an exam, if I had my arrow here and say, identify this cell, what would your answer be?
Melanocyte.
Keratinocyte, excellent. And what would this cell be? These are the easy questions, folks.
Keratinocyte.
And what's this cell up here? Keratinocyte, excellent. And keratinocyte, and keratinocyte, and keratinocyte. All of this stuff is keratinocytes. However, if you look closely, one of these things is not like the others. Remember, one of the things that we said about melanocytes is that while they produce the melanin to give coloring to the keratinocytes, they don't keep any of it for themselves. So notice this cell right here at the arrow has a very clear cytoplasm to it. Notice this one over here probably is a piece of a melanocyte that we cut off. Not a good example that we would use on the exam, but this one here is a perfect example. Notice that unlike every other cell we see on here, this one has a very, very clear cytoplasm, and that makes it very easy to identify. So histologically, on a microscope slide, you would be responsible for clearly identifying keratinocytes. That's the easy one. But you will also be responsible for identifying melanocytes. Because of their clear cytoplasm, they also are very easy to identify. Okay? Questions on that? All right, excellent. So again, and let's do it here because I want to make sure to emphasize it, and we'll use a red again. This single layer of cells right here is our stratum. Oops, sorry, my mouse ran into my cord. There we go. So I had a little seizure, but other than that, we're good. Is our stratum basale. Single layer of keratinocytes, where we can find melanocytes and Merkel cells and dendritic cells. And as we talked about, as these cells divide and produce new cells, they shove the old cells up. And as they shove the old cells up, they shove them into the second layer of our epidermis, or sublayer, let's be specific, sublayer of our epidermis, and that is the stratum spinosum. Stratum spinosum, as we see here, is typically comprised of somewhere between 8 and 10 layers of keratinocytes. These keratinocytes in this area, and again, let's go ahead and use the highlighter again to emphasize this region. So again, all of these. There we go. A little bit of that there, a little bit of that there. Perfect. Somewhere between 8 and 10 layers. And notice we have an undulating bottom to this. So again, the two layers do not just lay flat on each other. They interweave with each other. But we can still see, so it varies in how many layers they are. But these cells are the newly divided keratinocytes. Remember, keratinocytes division, really any cell division, requires a tremendous amount of energy. So what we have here are cells that are no longer dividing. And that means they have some energy to be able to do other things. Now, of course, being keratinocytes, what they want to do is to make keratin, to make those lamellar granules, to make the proteins and fill themselves up. However, before they do that, and what they do here in the stratum spinosum, is they actually are primarily involved in forming cell junctions. In particular, they are forming desmosomes. What kind of cell junctions were the desmosomes again? What do desmosomes do for our tissues? I'll wait. Again, there you go, connect things together tightly, right? Prevent shearing, excellent, perfect. Those are the things I like. These are the spot welds that really tightly hold these together, right? We want the skin to be that protective barrier, so much so that, like we said, when these cells die, when they're damaged, when they're burned, they stay together and peel off in sheets. In fact, these desmosomes is where this layer gets its name. If you think about it, let's cheat. Here are these kind of cuboidal and columnar shaped cells. Now, as we've mentioned, when we're processing this to look at it under the microscope, we cut a chunk of the skin out, we freeze it, we slice it with a knife, we put it in all these chemicals and process it. And part of that process, it causes the tissue to dehydrate. And as it dehydrates, water goes away and the cells shrink. Right? However, what happens with these cells, and I'm going to go ahead and change my drawing color. Let's go to orange. This cell forms a lot of desmosomes where it connects them to this other cells that surround them. And so what happens is when this cell starts to dehydrate, it shrinks, but as it shrinks, it still stays attached to the cells next to it. And so as it shrinks and still stays attached to the cells next to it, notice it starts to give the cell these kind of pointy or spiny appearance. So the cells in here have a spiny appearance because of all the cell junctions that are holding them together. So that's what we have here in our stratum spinosum. In our stratum spinosum, we have these cells that are forming desmosomes between the cells, forming those tight junctions to resist those shearing forces, to connect them together tightly and strongly. And that's where this layer gets its name. All right, giving it its flexibility and its strength. Now I want you to notice one other thing as well. Notice all of the, and let's get my drawing again. Here and here, these first two layers, and I know I exaggerated it, so there we go. In the stratum basale and in the stratum spinosum, as we said, it's all keratinocytes, but the cells are, as I just mentioned, cuboidal and columnar in shape. They're not flat yet. But remember, that's okay. With a stratified tissue, we just identify it by the shape of the cells on top. I mention it here, though, because notice as we go to our third sublayer, here in the third sublayer, the stratum granulosum, now the cells are finally starting to get squamous in shape. Now they are flattening out. And again, the key to a lot of these layers are in their names. granulosum. These cells, notice, are very dark and deep in color. I'm going to go ahead and write this right on top of it. This is a dark colored, whoops, hold on, layer. And the reason that it is this dark colored layer is that it has these granules. This area, the stratum granulosum, is now finally where the keratinocytes get to do what keratinocytes want to do. This is where we're doing our major protein synthesis. Oops, and that one I do need to change the color. We're doing our major protein synthesis in this area. These are the cells that are producing the keratin, that are producing the lamellar granules. All right, so clear that. Here we go, that. So again, this is where massive protein synthesis is taking place. And again, remember, they're called keratinocytes, but really they produce two things. They produce those keratohyalin granules, which are put together to form the keratin bundles. And that's why these keratin bundles love stain. So typically pretty much any tissue stain you use, they absorb massive amounts of it, which is what makes it a nice, dark, distinct layer, very easy to be able to identify. But remember, they're also producing those lamellar granules. And those lamellar granules are actually being released from the cell, producing that waterproof sealant on the outer surface. This is the turtle wax that is going to hold all these cells together and make the skin waterproof. Now, as we mentioned, these cells are super busy and super active, but they're almost too busy and too active. The cell fills itself up with keratin. The cell seals itself with a waterproof seal to the cells around it. And essentially what the cell says at that point is, you know what? I am so filled up with keratin, I don't really need this nucleus. I don't really need these organelles anymore. And it basically chucks them. The cell basically undergoes a programmed cell death, where the cell basically kills itself. They have a fancy name for that process. What do we call the process where a cell undergoes? There you go. That's the magic word I was looking for. Absolutely. After these cells fill up with keratin, after they seal themselves to the cells next to them, they undergo a programmed cell death, a process called apoptosis. And basically what that means is every single cell from that point up, all of these cells from this point up to the very top of the skin are now all going to be dead keratinocytes.
Question?
Yes.
The previous layers, we said that they're keratinocytes. So aren't they dead already in the deeper layers before this?
No. So all of the cells from this point down are living keratinocytes. All of the cells from this point up are dead keratinocytes.
What's the difference between the living and the dead? Is it, why are they're called the keratinocytes, but they still have the nucleus in them?
Yeah, again, they're called keratinocytes because they make keratin, right? That is their job. Their job is going to be to make keratin. But these cells fill themselves up so much with keratin that basically they chuck their nucleus, they chuck their organelles, and to answer the question, basically it's just released into the interstitial fluid of the tissues and will get broken down and reabsorbed. But yeah, and the cells basically die. They undergo this special programmed cell death. So basically up to that point they're alive, and after that they are dead.
So at this point they're still alive, correct?
Yes, in this layer, in the stratum granulosum, they are still alive. However, as they leave the stratum granulosum and go superficial, as they leave, they are now dead. So basically this, and let's go ahead and use my highlighter again. We'll use green this time. Basically this barrier right here, again, my line is pretty thick. So that is the stratum granulosum. That layer down, from here down, everything from here to here, all of these are living cells, and everything above it are dead cells. Okay, they're still cells, just in two different states. Here below the line, from the line down, those cells are alive. Above the line, the cells are dead. Okay? Excellent. Now, obviously, Can dead cells do a whole heck of a lot?
No.
No, but they are hardened cells that are fused together. So basically everything above this, their job is just to be that protective layer. But they certainly can't divide. They certainly can't make proteins. They cannot do anything else. They're basically just dead cells providing protection, right? Now, here's where things get interesting. Notice here in this one, and I'll go ahead and use my highlighter. There is this layer right here. This layer right here is the stratum lucidum. The stratum lucidum is the special layer that is only found in thick skin. And where do we say the thick skin was again?
Palms and
Palms of the hands,
bottom
soles
of
of the feet. Excellent.
yeah.
Yeah, absolutely. Right. So in those areas, this is the only place where we have the stratum lucidum. In thin skin, like the forearm or the scalp or a face or any of those areas, it just goes straight from the granulosum to the stratum corneum. So this layer is only present in the thick skin. Now, it does bring up an interesting point, though, because this green line that I've drawn here is dead keratinocytes. All of these cells in these layers are also dead keratinocytes as well. And as we mentioned, can dead keratinocytes really do anything? I mean, they don't, other than being a protective layer, they don't do anything. They don't have a job other than being that protective layer. So some of the questions have come as to why are these divided into two different layers? It's not like this layer does something different from this layer. But what's interesting about this stratum lucidum, and some people, remember we talked about artifacts, some people believe that it's just an artifact of the processing. But what has been found is it is consistently found in pretty much any stain that is used. And so while it doesn't have a different purpose, it is a distinct layer or sublayer. So it is a distinct region. So we do identify it as a specific sublayer. All right. Now, again, the name is the stratum lucidum, and it is called lucidum because it is a clear layer. Notice when we look at this picture, this layer doesn't look particularly clear. However, this is the new picture from your textbook. This was the old picture from your textbook. Now, for whatever reason, again, your textbook actually used one that had a gland going through the center of it, disrupting the layers. So that made it a little bit confusing. But here, you see where it actually gets its name. In this particular stain, as you can see, it is a very, very clear looking layer. Right? If something is lucid, it is clear and understandable. So this is a very clear layer, that stratum lucidum. It's relatively small, just 2 or 3 layers of dead flat keratinocytes. And again, no nuclei, right? Stratum granulosum, very dark, lots of nuclei below this line, but again, above this line, no nuclei. These are dead, clear cells in this stratum lucidum.
Question?
Yes.
So if somebody would ask, what's the difference between this layer and the layers that, you know, are more superficial, besides the look that it's clear, what, is there any other differences?
Nope. This is deeper and it is clear in color, but other than that, they're just dead keratinocytes. So, and I guess the size, this is 2 or 3 cell layers thick, whereas the stratum corneum above it is much thicker. So there's a difference in size and it's deeper. But other than that, yeah,
same function, no different functions or anything like that.
Yep, correct.
Okay, and the location, of course, it is different because it's only in thick skin, correct?
Also correct, yes.
Okay.
Yep, perfect, excellent. And that brings us to our last layer, the stratum corneum. This is the most superficial layer. Notice I've got written here 20 to 30 layers of those dead keratinocytes, but the key is this is the average. It varies dramatically throughout the body. Where, and I think I've already given you this information, but I'll ask it again. Hold on, does the lucidum not have melanin? Great question. No. In fact, melanin basically is only found, and I'll use my highlighter again because it's convenient. The melanin basically is only found in the deeper layers. So melanin is just found in the deeper layers, because remember, that melanocytes projections only come out so much. And so melanin is only found in the stratum basal and the stratum spinosum. So in the deep layers of the stratum spinosum. So you're only going to see the melanin down in those layers. So yeah, by the time the cells get up here, the melanin is gone. All righty. Now, oh, I erased my average too. So I think I've mentioned it. Where is the stratum corneum the thickest in your body?
Bottom of your feet, the
Yeah,
soles.
and more specifically, the heel, right? The heel
The
of
heel.
your foot. In the heel of your foot, you have over 200 layers of keratinocytes in your stratum corneum, right? So you can use that pumice stone and work away at it. Where do you think it's the thinnest?
Your face?
You're close. Can you be a little bit more specific? Lips is a good guess, but no. Face, definitely, we're in the right area. Lips is a good guess, but
Eyelids?
eyelids. There you go. Exactly. In your eyelids, the eyelids is your thinnest skin. In your eyelids, you only have about 2 to 3 layers of keratinocytes in your stratum corneum. So there you go. So your thinnest skin is your eyelid. Your thickest is the heel of your foot. But if you take the whole range of everything together, the average is about 20 to 30 layers. All right. And again, once they get to the top, then these cells at the top are what slough off and become the dust on your computer screen and your kitchen countertops and all the other things that dead skin cells become. Right? Now, one of the interesting things about the connections, while these are dead cells, one of the things that does happen as you put pressure on the skin, the attachment between these dead cells will enhance, will come together, will become stronger. What do we call that when that occurs? When we increase the attachment of these dead cells? I know you know, because maybe you spent a summer digging ditches. There you go, a callus. Absolutely, you get that callus on your finger or on your hand from doing work or other areas. That callus is that increased attachment of layers from pressure being put on it. However, you have to be careful because if instead of pressure, what happens is maybe you put on a new pair of shoes and you go for a 15-mile walk, and as you're doing that, it's constantly rubbing back and forth against the layers. And as it's rubbing back and forth against the layers, it may cause the sublayers of the epidermis or the epidermis and the dermis to separate from each other. And what do we call that condition when the layers separate? Typically, when they separate, that area fills with fluid. And what do we call that condition?
Blisters, or?
You call it a blister. There you go. Exactly. You call it a blister. Exactly. So blisters form when the sublayers of the epidermis separate or the epidermis separates from the dermis. And basically, the way to tell which way it is what it's filled with. If the blister is filled with a clear liquid, that's the interstitial fluid. And so that's basically when the epidermis layer slips. When the epidermis layer separates, it fills with interstitial fluid and you get that clear fluid. However, with a deep blister, or what we call a blood blister, that's typically where the epidermis and the dermis separate from each other. Remember, the dermis has blood vessels. So when you separate there, then that space can fill with blood, and that we call a blood blister. All right.
Quick question.
Yes.
When I was doing the homework, so the epidermis has 0 blood and 0 nerves, correct?
No, it has 0 blood. It has a ton of nerves.
Because one of the question was that if you cut it, you don't feel any pain. So if he has the nerves, how do you feel pain, or you don't feel pain?
You don't feel pain if you cut yourself?
No, it was talking about, one of the question was like if you peel the skin from your heel or something like that,
Okay. Yes.
that you don't feel the pain.
Okay, true. The superficial layers of the skin, like the stratum corneum, do not have nerves in it. Remember, as we've talked about, and I'll draw it because remember, you're not responsible for it histologically. We have talked about how there are those tactile cells located deep in the skin. There are going to be other nerves we'll talk about, like, again, we haven't talked about them yet, but we will. Free nerve endings. These free nerve endings are what give you your sensation of temperature or or pain or things along those lines. So there are nerves in your epidermis, but they are deep. So yeah, if you take that pumice stone and while you're in the shower today and you peel off the first 15 layers of your those 15 layers of the skin on your heel, are you going to feel that? No. If you take that pumice stone, and again, I'm not encouraging you to do this, but if you took this pumice stone and you rubbed it on the surface of your face or even your eyelid, would you feel that?
Yeah.
Yeah, absolutely. So yes, and these superficial layers, especially in the thick skin like this, are indeed, there are no blood vessels and there are no nerves. But there are nerves in the deep layers of the epidermis. We definitely receive a lot of sensory information from our epidermis. Okay. Great question, Nicole, and that's exactly the question I was going to answer next. So this whole process, and again, notice we went from the histology to a pretty picture that shows this. I like this picture for a couple of reasons because it really emphasizes these processes. Here we see the stratum basale or stratum germinativum undergoing the process of mitosis and division. Notice also we have our Merkel cell, that tactile cell. We have our melanocyte with its processes. We even have that dendritic cell migrating through. As this divides, it produces these new cells, and these cells are forming the desmosomes. We see these spiky cells connecting them together in the stratum spinosum. Then they elongate and flatten and get into major protein synthesis in the stratum granulosum, filling up with keratin, fusing themselves together so much so that they then undergo apoptosis and die, and in thick skin forming that stratum lucidum, moving up to the stratum corneum, and then ultimately sloughing off. This process of going from cell division to protein synthesis takes about 2 weeks. Then, and obviously depends on the part of the body, but from the part of the body, from dead cell to dust on your countertop takes about 2 weeks. Now, obviously, it happens much more rapidly in the eyelids than it does on the heel of your foot. But in general, basically, every 4 weeks, right, every 28 days, you have a completely new epidermis, right? And again, you don't necessarily think of it in those terms, but you know you're aware of that because about a month ago was Mother's Day. And to let mom know you really love her, right, you decided to take not just any marker, but a permanent marker and put mom on your arm and put a big heart around it. And you promise not to wash your arm, right? And you're not going to wear any shirts with sleeves so that it will stay there forever. And will it really stay there forever? No, even if you haven't bathed, even if you haven't worn a shirt for 4 weeks, it's going to be gone because those cells sloughed off. Instead, if you want to let mom know you really care, what do you need to do?
Do your chores.
You need to not put that ink on the surface. You need to go through the epidermis all the way down to the dermis and inject the ink there, right? Get that tattoo. That one's a little bit more permanent. So this process, there we go, of keratinization and growth, right, takes about 2 weeks, like we mentioned, to go from stratum basale to a fully keratinized, fully filled dead cell, about 2 weeks, and then about another 2 weeks for it to migrate to the surface and slough off. And again, oh, go ahead, yes.
So it does matter on your age also. Doesn't your cells start to slow down? Like
True, and not only does it slow down, but typically the cells, the number of cells will decrease as well. So as we age, the cells, the thickness of the skin decreases a little bit along those lines as well. So yes, they become a little less metabolic, and there typically are fewer layers, making it more delicate. Yeah, absolutely.
Question?
Yes.
For the thick skin, the one like at your heel,
Mm-hmm.
I'm assuming that it doesn't completely go through 200 layers in 4 weeks, correct?
No, like I said, so again, these numbers are averages, obviously, yes. The thinner skin, it'll go through faster. The thicker skin, it'll take longer, absolutely.
Okay.
Yeah. And this is all just under normal homeostasis as well. The other thing that can happen is that if the skin becomes injured, then we can grow the skin back faster. When skin gets damaged, when skin gets irritated, what happens is our keratinocytes produce a protein that acts kind of like a hormone. It's not truly a hormone, but it is a hormone-like protein. And that hormone-like protein is called epidermal growth factor. An epidermal growth factor targets the stratum basal cells, making them divide more rapidly so that we produce skin more rapidly and that skin grows back more quickly. Now, notice I said irritation to the skin. Obviously, if you're riding your bike, you fall off and you scrape your knee, you've injured yourself and you're going to want it to grow back faster. But what happens is any irritation to the skin can cause this, right? You do that chemical peel because you want to remove those thick, coarse stratum corneum layers and have a nice shiny surface to your skin. Well, guess what? That's irritating the skin. And ironically, you're doing that so that you get that nice smooth looking skin, but it stimulates the skin to grow more cells back faster. When you shave, the irritation of the razor stimulates more skin growth. And as we'll learn, hair grows in the exact same way that skin does. So the irony of shaving is when you shave your skin, you shave the hair off of your skin, it irritates the skin produces that epidermal growth factor, and it makes the hairs grow faster. All right. So we have that going on. And so again, we have that ability to be able to produce it more rapidly as needed for healing processes. All right.
Quick question.
Yes.
Sorry, this might be maybe a stupid question, but
The only stupid question is the question not asked.
okay, you mentioned shaving, right?
Mhm.
Now, what about if you do like waxing? Is that the same thing?
Sure, it
as
irritates the
shaving?
skin. Well, it irritates the skin, but it has a different effect on the hair. So when we talk about the hairs, we'll come back to waxing. Let's talk about waxing when we get there.
Okay.
Okay? Yeah, because it'll make more sense when we talk about the hairs. All right. Any other questions? All right, excellent. So again, and let's do this for a second. Let's go all the way back to this picture. This is our skin. And remember, as we just finished talking about this right here, highlighter, from here to here, this much of the skin is the epidermis. And as we just talked about, in the epidermis, there are 5 sublayers. Well, if you think about the skin, that is all the epidermis, but all of this down here is the dermis. And if we can fit 5 sublayers here into our epidermis, how many possible sublayers could we put here inside of our dermis? Anyone know how many sublayers we have to our dermis?
5.
Yeah, it's just 2. So it's not as scary. But if you think about it, again, if we think of the relative size of the epidermis versus the dermis, it could be a little intimidating when we first look at it. But no, as it turns out, the good news is there is only 2. Right, and oh, conveniently enough, they actually point them out here. Papillae layer and reticular layer. Now, remind me again, the epidermis was comprised of a keratinized stratified squamous epithelial tissue. The dermis is comprised of connective tissues. And what connective tissues again?
The
Areolar
tissue.
Areolar and?
was it the loose?
No,
Right.
the areolar is the loose one. What was the other one? Areolar and
Dense.
dense.
That's irregular.
Dense irregular connective tissue. Excellent. Dense irregular connective tissue and areolar connective tissue. Notice the dermis is made up of 2 tissue types, and the dermis also has 2 sublayers. Right? Think that that's a coincidence? It isn't. The superficial layer, the papillary layer of the dermis, is the one that is made of the areolar connective tissue. And the reticular sublayer of the dermis is the one that is made of the dense irregular connective tissue. So in our dermis, we are going to have two tissue types. We are going to have two sublayers. Excellent. So let's jump back to that part and start talking about our dermis. Oh, let's go. I wanted to show one more thing too. This was the one example of the thin skin that we've been looking at, just again to emphasize the point. How many sublayers in thin skin?
1.
No. How many
4.
sublayers in the epidermis of thin skin?
4.
4. Excellent. Stratum basale, stratum spinosum, stratum granulosum, and stratum corneum. Remember, with our thin skin, there is no stratum lucidum. So it goes straight from granulosum to the corneum. So there's no lucidum here on this one. Whereas obviously our thick skin has that lucidum in between the granulosum and the corneum. All right, excellent. Perfect, excellent. All right, so let's talk about the two sublayers of the dermis. The first sublayer is the papillary layer, and it's named that because of the relationship between the dermis and the epidermis. As we've talked about, these layers do not just lay flat on the surface of each other. Instead, they interdigitate with each other. There are long finger-like extensions that stick up of the dermis, and the name for a finger-like extension is a papillae. Papilla being singular, papillae being plural. And these finger-like extensions, these structures, are where the papillary sublayer gets its name. Now, why would we want the dermis and epidermis to interdigitate? Why not just have them flat on each other? What's the advantage of having them interdigitate? Excellent. Part of it is definitely a better connection, right? If you put your hands together flat, and even if you push really hard, you can still slide your hands pretty easily back and forth against each other, right? And is that what we want our dermis and epidermis doing, sliding against each other? No. But if we interlock them, then it holds them together much more tightly. But there's another important reason to have them interdigitated like this as well. What's the other important reason?
Is it to separate the environment?
No, not necessarily to separate it. Why do we put microvilli on the surface of a cell again? Why do we put surface area, increasing surface area? Remember, our epidermis is avascular. All the oxygen, all the nutrients that it's going to get, it has to get from the blood vessels of the dermis. So by interdigitating them, we increase the surface area, which makes it easier for the dermis to give the oxygen and the nutrients it needs to the epidermis. So it holds them together tighter, and it increases the surface area for better diffusion of materials. Excellent, 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 excellent. Perfect. Now, the papillary layer is the superficial 20% of the dermis. And as we mentioned, it is the one that is made of the areolar connective tissue. As someone pointed out, remember, this is our loose tissue. And because it is a loose tissue, there's lots of space for lots of stuff. One of the main things we need in here, and we see this nicely on the illustration, are lots and lots of capillaries, because again, we need to be able to provide the oxygen and the nutrients necessary for our epidermis to be able to grow. But this is also a place where we have a lot of sensory structures. Here, like for instance, these, and I'll highlight them in purple. This one here and this one here are structures that are what are known as Messner's corpuscles, right? Of course, they're called Messner's corpuscles because, as we know, good old Bob Messner was the first person who ever identified them, planted his flag in it, put his name on it. but they're also called tactile corpuscles. And they're called tactile corpuscles because they provide the same type, let's say it this way, same type of information as the tactile cells. Now remember, tactile cells are found in the epidermis. These tactile corpuscles are found in the papillary layer of the dermis, but they provide the same type of information. And what type of information did we say those tactile cells provided? Excellent, that fine touch, that fine discrimination. Now, one of the things, remember, that we talked about with our tactile cells is that they're not equally distributed throughout the body. Remember, tactile cells are most dense in the palm of the hands, especially the thumb, the face, especially the lips. Tactile corpuscles are also not equally distributed throughout the body. They actually are more common in areas where the papillary ridges become bigger, and that is in the thick skin. So tactile corpuscles are more common on the palms of the hand and the soles of the feet. All right, so the soles of the feet have more tactile corpuscles. The face has more tactile cells. The hands have more of both. Which again, if you think about it, makes sense. How do we primarily explore the world? Do you explore it with your elbows? No, we explore the world with our hands. We have massive number of those fine discrimination cells here in our hands, both tactile cells in the epidermis and tactile corpuscles in the dermis. All right. I'll get rid of that, and that'll allow me to do this. But there are other types of nerves I mentioned as well. One of those, as I mentioned, are free nerve endings. Free nerve endings are just, oh, that's what I need. Free nerve endings are nerves that basically don't have a specialized structure at the end. And they give us general sensations. These free nerve endings basically provide general sensations. General sensations, things like pain, things like itch, things like tickle, things like temperature. Now, I know when we think of temperature, we don't think of temperature as a general sensation. But can you right now take your finger, stick it in your coffee, and tell precisely what temperature it is? Yes. Yes? You can tell precisely to the degree what temperature it is. That would be impressive. Most people cannot, right? In fact, temperature really is a change in temperature, is really what we perceive. We don't actually typically perceive absolute temperatures. We perceive changes of temperatures, right? You probably did this in elementary school, but if not, or if you want to experience it again, this is something you can do today at home. Do it with your kids, with your loved ones. You set up 3 bowls of water. One that is warm water, not hot, not boiling, warm water. One that is ice water, and a bowl that is room temperature. You put your hand in the warm water, you put your right hand in the cold water, and you let them sit in there for 30 seconds. And then you take both hands and put them into the room temperature water. And when you do that, does the room temperature water feel the same to both hands?
No.
No, because what we actually measure are changes in temperature. That's how we perceive temperature. It's a general sensation. Think of it this way. If you were sitting in a hot tub and I turned that up 1 degree, would you likely notice the difference?
No.
No. Now, let's take it a step further then. If you were willing to sit in a bowl, in a hot tub long enough, and every 15 minutes I turned it up 1 degree, would you be able to notice the change? Probably not. So does that mean I could actually boil the water with you in it and you would never get out?
No.
No. No, you're right. It wouldn't. You wouldn't notice the change, but here's what would actually happen. What's useful about us is, yes, you would not notice the temperature change. However, it turns out we have a second type of temperature receptor, And that temperature receptor is more of a pain receptor. So here's what would happen. I'd make it warmer, you wouldn't notice. 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 But eventually, we would reach a critical point where that temperature stimulated those pain receptors. And you would say, hey, this water is hurting me, and you would get out. Having that second temperature receptor is something that's actually pretty special. Not everybody has it. Frogs, for instance, don't have it. So again, not encouraging this to be a weekend activity for you, but technically you could take a bowl of water and put a frog in it, put it on a hot plate, and if you turn the temperature up slowly enough, 1 degree every 15 minutes, you could actually boil that water and the frog would never jump out. He would never notice the temperature change, and you could actually boil the water with him in it and he would never jump out. So yeah, so temperature is a general sensation. All right, now let's see, what's next? Oh, and then here's the electron microscopy picture from your textbook. Again, showing this areolar connective tissue with all of the loose space for all of the hunk and junk that is here inside of it. All right, now as I mentioned, this layer gets its name because of these dermal papillae, these finger-like extensions that stick out throughout the entire surface of the skin. However, in our thick skin and our thick skin only, these dermal papillae become enhanced, forming what are known as the epidermal ridges. And as hopefully all of you experienced, these epidermal ridges come together to form our fingerprints. Now, we call it fingerprints, but really, it is not just on our fingers. It is on the palms of our hands. It is on our toes. It is on the soles of our feet. These areas, these epidermal ridges become enhanced to help to enhance our gripping and our grasping, especially when wet, right? And again, you probably haven't thought of it in those times, but if you were sitting in that hot tub for an hour while I was slowly turning up the temperature on you, your skin would get pruny, right? We all talk about getting pruny from being in the water too long. But when we say get pruny, do you really get wrinkly all over? Does your forearm get pruny? Does your chest get pruny? No. Where do you get pruny? Palms of the hands, soles of the feet, right? Those are the areas where we have those enhanced ridges. All right. Now, what's interesting, and hopefully you guys got a chance to experience this if you took both yours and other people's in your family, they were probably similar. The reason for this is these epidermal ridges are mostly genetic. Let's say it this way, genetically determined. Mostly genetically determined. So since you share genes with the other people in your family, they are probably similar. However, if you have identical twins, do identical twins who are identical genetically, do they have the same fingerprints?
No.
No. So it's mostly, and again, mostly is that key word, most means not all. It is mostly genetically determined. And what's super interesting about this is your fingerprints, those epidermal ridges actually form in utero while you're developing in mom. So obviously the two babies are growing there inside mom, but they believe there's something about that environment. All right, one's on the left versus one's on the right, maybe slight differences in the chemical distribution, differences in temperature, whatever it is, there's something about those environments that make them different enough where their fingerprints will be unique. Now, they will be more similar than just two regular siblings would be, but they will not be identical. So while it is mostly genetic, it is not entirely genetic. All right, questions on that?
Is it possible just for a second to go back to the previous slide?
Sure, there.
Okay.
Although again, remember, you have these slides, so you should, I don't think this is a slide that have changed, so you should have all of these slides.
Okay, I'm good.
Yep, all right. Excellent. That brings us to our last sublayer. That last sublayer is the reticular sublayer, making up about 80% of the dermis. As we talked about, it is comprised of our dense irregular connective tissue. And here is that great electron microscopy view from your textbook, where you can see the collagen fibers at all sorts of different orientations within this tissue to provide, like we talked about, that resistance to multi-axial stress. All right. These collagen bundles and elastic bundles, these elastic fibers and collagen fibers provide strength, extensibility, and elasticity to the skin. Now, obviously, there are a ton of fibers in this tissue, but it's still a connective tissue, which means there's room for other stuff. And so there are going to be other stuff found in this area, as we see from the picture. For instance, the follicles where our hairs are going to grow are found in the reticular layer and then grow up from there. Our sebaceous glands, these are the glands, as we'll talk about, that are the oil-producing glands. What we think of as oil, but is really, as we talked about in the last class, is sebum, the substance that is produced from those. This is where we'll find our sweat-producing glands. Oops, I wanted that. Those sweat-producing glands, which are our sudoriferous glands, like the one right here. But also some sensory structures as well. Notice one of those sensory structures is this structure right here. Notice it looks, and I'll cheat and use my circle drawing to make a nice example of this. These are kind of elongated, oh, this isn't gonna work. All right, I'm just gonna cheat and draw it, try freehand. Layer upon layer, kind of looks like an onion, a cut onion, where we have all these layers, all these rings of material, of layers of plasma membrane. And these are, that's why they're called a lamellated layers corpuscle. Of course, they're also called Pacinian corpuscles. Why? Good old Bob Pacinian was the first one. Pacini was the first one who identified them. But what these layers of plasma membrane do is when they are squeezed, that stretches the plasma membranes and it sends an electrical signal to our brain. So these are the sensory structures that allow us to feel pressure, right? If you squeeze a tennis ball and you can feel that pressure, or someone squeezes your arm, or the pressure of your chair on your back and on your butt right now, that force that you're feeling, that pressure you're feeling, is from those Pacinian corpuscles that are found deep in the surfaces of our skin. All right. And then obviously this is where our large blood vessels and nerves are going to be located as well. All righty, questions on that?
Yeah, quick question.
Yeah.
For like your skin, for example, if you take a feather and just run it across your skin, that will be the tactile, correct?
Yeah, those would be so yes and no. The feel of it, the sensation you feel from it, where you're able to perceive where it's occurring in your body, would be the tactile cells and the tactile corpuscles. However, if it caused a confusing sensation that caused you to be tickled by it, those would be the free nerve endings. Right, that's kind of one of the interesting things about it. The tickle that would come from that is because those free nerve endings send a signal that confuses the tactile ones, and that confusion of neural input is what we perceive as a tickle. That's why it is impossible for you to tickle yourself. You can't tickle yourself because you are controlling the feather, and because you're controlling it, you know where the feather is, and it's impossible for that signal to be confused. In fact, they've actually done studies where you control a machine and that machine does the feather on you, and because you're controlling the machine, you still know you're controlling it, and so it is impossible to tickle yourself.
Okay, but it doesn't activate the laminated corpuscles.
No, because that's not because a feather lightly touching you, or even someone lightly touching you, isn't pressure. right?
Gotcha.
Pressure is deep. So if I grabbed your arm, you'd feel that. If I just rubbed you with a feather, that wouldn't be the Pacinian corpuscle.
Okay.
Okay? Excellent. Any other questions? All right, perfect. Now, I do want to talk a little bit more about the reticular layer, because we see this great picture from your textbook where it shows like completely random orientation to these bundles. That isn't entirely true. In certain areas of your body, the collagen fibers in your reticular layer will be somewhat arranged. Now, they're always going to be irregular, but in some areas, there's going to be trends where there's going to be more in certain directions than others. We can actually map this on the body. Here's the picture from your textbook that actually shows the general trends of the collagen fibers inside the reticular sublayer of the dermis. We call these the cleavage lines. Now, from a functional standpoint, if you think about it, these provide extra protection. If you think, for instance, in the joint here of your elbow, you're a lot more likely to have stress in your elbow in this orientation than this orientation. So it makes sense that the fibers are oriented in the direction where you're more likely to perceive stress. All right, so this helps to provide protection that way. However, from a clinical significance, as a clinician, as a nurse, as a doctor, why might it be important to know the lines of cleavage, to know the orientation of the collagen fibers?
Maybe when you do surgery and you cut somebody.
Exactly. If you do surgery and you're going to do surgery, for instance, on someone's leg, is it going to be better to cut across the collagen fibers or to make the incision with the collagen fibers?
With the collagen fibers.
With. That will dramatically decrease the scarring that will occur as a result of that. It will improve healing, right? And it will make it less likely to change the function or the integrity of it. Right. In fact, if you think about it, the whole field of plastic surgery, like especially like with the facial plastic surgery, is about studying these lines of cleavage, of how to make those incisions with the minimalist amount of scarring possible from them. So these these cleavage lines are important from a functional standpoint to protect our body, to protect our body with our skin, but we need to understand them from a clinical standpoint as well. All right, questions on that? Excellent. Just that easily then, we have identified the major organ of the integumentary system, the skin, identified the 2 layers, identified the 7 sublayers, 5 in the epidermis, 2 in the dermis, what's going on in those layers, what we find in those layers, and all of that. What we are left with then is talking about all of the accessory structures. We need to talk about the hairs, talk about the nails, talk about the glands. So we've got some of the heavy lifting done, but we still have a fair amount to do. So this is a good stopping point. I don't know how we are on time. Actually, we're doing really good on time. Perfect. Excellent. So it is 9:07 right now. So let's go ahead and take our first break, 15-minute break. So come back at 9:22, and at 9:22 we will restart. Yep. And I will start the recording at that time. Excellent. So I'm going to go grab my coffee. You grab whatever your stimulant of choice is, and we will meet back here in 15 minutes. Any questions? All right, I will see you shortly. So, like I said, we've talked about the dermis and the epidermis. So our goal now moving forward is to talk about the components of the skin. And the first one of those that we are going to do is going to be the glands. Now remember, as we talked about when we were discussing epithelial tissues, every gland that you are going to be responsible for in this class, and not just this section, but really from the end of 430 to all through 431, there are going to be several things you are going to need to know. Obviously, you're going to need to be able to identify the gland, know it by name. We need to know our structural classification. We are going to need to know our functional classification. And remind me again, how many structural classifications did we identify? I did turn the microphone on, right? Anyone remember how many structural classifications we identified for glands? I'll wait if you have to look back at your notes to figure it out. There you go, excellent. Technically, absolutely. Technically, we identified 8. However, remember, one of those 8 was unicellular, right? So all of the glands we're gonna be talking about of the skin, none of them are unicellular. So if you think about it, if we take that unicellular one out, then that leaves us 7 possible structural classifications we're gonna use. And how many functional classifications were there?
That one was 2, correct?
Well, technically 3, right? There were technically 3, but you are correct in that, remember, the epicrine mode of secretion is something that we said is not found in humans. So you are absolutely correct that when it comes to, again, thinking about for the answers to these questions, there are basically going to be 2. It's for every human gland, it is either going to use the merocrine mode of secretion or the holocrine mode of secretion. So that does make that easy that way. So we needed to know, be able to identify it by name, recognize that we need to know its structural classification, we need to know its functional classification, we need to know the substance it produces, right? And because we are talking about the skin, we'll also need to know the locations in the body, locations in the body where these found. And by locations, I mean specifically the regions of the body. I don't mean sublayers of the skin. Right, so I don't want you to say that a sebaceous gland is found in the reticular sublayer. The reticular sublayer is where pretty much all of them are found. Right, what I want is the region of the body, like the brachial region or the axillary region or the buccal region or whatever it is that way. So the region of the body. So these are the things that on an exam, I will show you. Oh, that's what I forgot to do. Hold on one second. I think I have this easily available. If not, we'll do it after the next break. All right, I'll have to find it during the next break. I don't have easy access to it right now, so I'll have to figure out where I put that. All right, so let's go back and do this. Perfect. All right, so those are the information we're going to need to know about all these glands as we move forward. All right, histologically, I could show them to you, models, pictures from your textbook, things along those lines, and you need to be able to identify these 5 basic facts about our glands. So let's start that first by talking about our sebaceous glands. Sebaceous glands are the ones that are oil-producing glands. When we talk about the location, it's a little easy for the sebaceous glands because they are located pretty much on every surface of your body except the palms of your hand and the soles of your feet. What's special about the palms of the hands and soles of the feet again?
They're thick skin.
They're thick skin, absolutely. That's one of the things that's special about it. And what's also special about those 2 regions of the body?
He has no hair.
No hair, exactly. All sebaceous glands are actually connected to, or really, if we think about it, they actually release their secretions into hair follicles. So they actually release their secretions into the follicles of the hair. So areas where we don't have hair, like the thick skin, there are no sebaceous glands. So you have no oil-producing glands on the palm of your hands, on the soles of your feet, because you have no hairs there. All right? So here we see a pretty picture from your textbook where we see this sebaceous gland attached to a hair follicle where it is going to release its secretion. As I mentioned, we need to know the structural classification and the functional classification of this. The structural classification is a branched acinar. So remind me again, or again, it could also be branched alveolar. Both of those terms are acceptable. Remind me again what that means to be a branched alveolar gland. When's the test again? Saturday, Sunday, 3 days from today? Again, we spent a lot of time on glands. You should definitely be expecting an essay question on that. Excellent. Branched alveolar. Branched means that it has one duct, and it is going to have 2 or more secretory structures. And what do we know about those secretory structures? Round, perfect. So, oops, that's not round. Absolutely. Basically, it is one duct with 2 or more ball-shaped secretory structures attached to it. That's what a branched alveolar or branched acinar means. And we do see that a little bit here with our cross-section. This structure here, hold on, I don't know if you can see the eraser while I do that, so let's switch to this. This is actually the hair follicle. This is where the hair would be located. We'll talk about the anatomy of that in just a little bit. But what we can see here is we can actually see the duct where it is actually releasing its contents into the follicle of the hair. And then here we see the multiple round ball-shaped secretory structures. Now notice something about these secretory structures. Notice they're very clear in color, kind of like adipose like we've seen before, or also kind of like mucin that we've seen before as well. However, notice there is a big difference. Here we can clearly see the nuclei of them, and unlike adipose where the nucleus is shoved to the side, here the nucleus is smack dab right in the middle of the cell. And the reason these are light in color is because, like adipose, they are producing an oily substance that they're going to be releasing. And as we already mentioned, the substance that they're going to be producing and releasing, and I'll write it down here even though we're going to say it again in a second, is sebum. It is that oily, viscous secretion that is being released from these glands, from these branched alveolar glands. Its functional classification, or again, we could also think of this as mode of secretion, right? How does it release its substance? Is holocrine. What did holocrine mean again? What does holocrine mean? Well, we'll talk about its function in just a minute, but what is the holocrine? There we go, excellent. Cell fills up, divides, fills up with its substance, and the cell ruptures, dying, releasing all of its contents. Remember, it produces a very thick, very viscous, very organic secretion as a result of that. Excellent. And like we said, it is going to produce sebum. Sebum is that oily substance that you get on the surface of your skin. It moisturizes and waterproofs the skin. Again, as I mentioned, now that we're all washing our hands 17 times a day, your hands often dry out as a result of that. The skin can become cracked as a result of that, so it helps to moisturize and waterproof it. It actually has a minor antibacterial substance. Now, is the goal, and again, this is one of those tricky things, is the goal to be completely devoid of bacteria? We want our skin completely devoid of bacteria. Is that what our goal is? Is that what we want? No. In fact, in a single square centimeter of your skin, you can have as many as 10,000 bacteria. Now, I know as soon as I say that, most people go diving for their Purell, but what you have to remember is most of that bacteria is what we call resident bacteria. And that resident bacteria is actually beneficial to us. It is benign. It doesn't cause damage. And what happens is if a harmful bacteria comes to the surface of our skin, the resident bacteria is able to outcompete it for resources, stop it from getting purchased, stop it from growing. When we're constantly using all this antibacterial stuff, we're wiping the slate clean so that any bacteria, good or bad, has an increased likelihood of being able to make purchase on the surface of our skin. So again, bacteria isn't necessarily a bad thing. There are certainly bad bacteria, but there are also good bacteria as well. Right? As we talked about, the secretion of the sebaceous glands are controlled by hormones. And so when we especially enter puberty and there's a dramatic increase, that causes an increased production of sebum. And when you have all this thick organic material that is just leaving out of a single duct, then that single duct can become congested. It can become blocked. And as we talked about, this is thick, viscous, organic, warm, moist material, just the kind of thing bacteria love. And so that can become infected. And as we talked about, that becomes acne. All right. Questions on that one? That one's pretty simple and straightforward. All right, let's talk about the next gland, sudoriferous glands. Now, this is where things get a teeny bit trickier. Sudoriferous is a general class of glands. Obviously, if there's a general class, that means there are going to be specific types. And in fact, there are 4 specific types of sudoriferous glands. All right. Sudoriferous glands are our sweat-producing glands. And all 4 specific types have the same structural and functional classifications. All right. So for all of our sudoriferous glands, they are all going to be simple coiled tubular glands. Remind me again what that means to be a simple coiled tubular gland.
One layer.
Well, you got one, so there's one duct. And then what else? There you go, excellent. So one duct, and technically the duct is actually typically straight, although it can be coiled as well. But you got the right idea. One duct, one long tube-like secretory structure, but that secretory structure is going to be twisted and turned around. Notice that's what they've drawn here. Notice in our illustration here, we have this nice illustration where they've left it 3-dimensional, so we can see how that one tube coils around. But remember, typically we're going to be looking at this under a microscope. And when you look at this under a microscope, you have to cut through it. Like we talked about in the last class, if you have a ball of twine and you cut through that ball of twine, you don't see one piece of twine that's spinning around. You see a bunch of pieces of twine. And so when you cut a simple coiled tube, it looks like this illustration over here, where we see lots of pieces of the tube, some of them cut in cross sections, some more longitudinal, some oblique, cut at all these different angles. They've done a nice job of showing this to us in the illustration, but we can also see it when we look at it under the microscope as well. So here under the microscope, we see this cut pieces of tube all clumped together. And so again, it doesn't look like a single structure, but it is a single coiled tube that has been cut. All right. Oops. So all of our 4 types, let's do this. I want that back, and I want to erase this and this. Perfect. So 4 specific types of sudoriferous glands, but all of them have the same structural classification. They're all coiled tubes. They all use the same functional classification, or they all use the same mode of secretion, merocrine. And what did merocrine mean again? Exocytosis. Excellent. They use exocytosis. Perfect. Excellent. So that is true for all 4 types. Again, remember this because it's about to get confusing. The most common type of sudoriferous gland are what are known as the eccrine sweat glands. These eccrine sweat glands used to sometimes be referred to, and every once in a while you'll still see it in the literature, where they will be referred to as merocrine glands. However, remember, all sudoriferous glands use the merocrine mode of secretion, so it isn't necessarily a useful way to describe them. Instead, we will use the term eccrine. Eccrine is the appropriate term for them. These are the most common, and these are the ones that produce watery sweat. All right, this kind of sweat that is used for regulating our body temperature. Here is what they look like histologically. Again, we can clearly see that they are coiled tubes when we look at it, but I want you to notice something else about it as well. Here, and again, I'll use the highlighter to do this so I don't obscure it too much. We'll use green for this. Notice here is the wall, those simple cuboidal or simple columnar cells, and we see the size of the wall, and look at the size of the lumen. The lumen on these are very small. So notice the wall is much thicker than the lumen, which if you think about it, makes sense. These are producing a watery sweat. And do we really need a big, huge lumen to produce a very watery secretion? No, probably not. Now, let me ask you another question. Where on the body are these located? You guys can't give me one location in the body where eccrine sweat glands are located?
Epidermis.
Again, remember, region of the body, part of the body. I don't want layers. Axillary region, excellent. Anywhere else?
Inguinal region.
Inguinal region, anywhere else? These produce watery sweat. Tell you what if you were to get up and do 75 burpees in your room right now and then sit back down? Would you be sweaty as a result of that?
Yes.
Yeah. And what parts of your body would be sweaty after you did that? Or let me ask a better question.
Everywhere.
Would there be a part of your body that wasn't sweaty after doing that? Would there be any part of your body that wouldn't be sweaty after doing 75 burpees? No, it'd be everywhere. So guess where eccrine sweat glands are located in your skin?
Everywhere.
Everywhere, absolutely, every surface. So when I ask you for a region of the body where an eccrine gland is located, is there a wrong answer to that? No, because they are literally everywhere. Every surface of your body has them. Now, they're not equally distributed. For instance, you have more of them in the palm of your hand than you do on your forearm. That's why your palms get all sweaty when you get nervous and things like that. But every single surface of your skin is covered with these watery sweat glands. All right. Questions on that? All right, excellent. So then let's talk about, so again, whoops, hold on, let's get rid of that. These produce watery sweat. The second type, oh, and again, oh, this is great. So they produce our watery sweat. If you stay inside in a controlled environment with your air conditioner on, they produce on average about ½ liter of sweat during the course of the day. However, if you were to do 75 burpees, as we talked about, or as it's nice and getting hot outside, if you decide to go weed your backyard or tar your roof, they can produce as much as 8 liters of sweat in a 24-hour period of time. Now, do you have 8 liters of excess fluid in your body you can lose in a 24-hour period of time? No, it's one of the reasons why it is so vitally important in these Sacramento summers to stay appropriately hydrated, because they have the potential to produce and release massive amounts of water from your body to help to regulate body temperature. All right. And again, it isn't just water, it's antibodies, it's ions. As we said, this is a way that toxins and other things can be released. We talked about sweating out your alcohol, sweating out garlic. If someone has uncontrolled diabetes mellitus, Glucose can actually be sweat out. Their sweat will take a sweet smell to it. It's one of the indicators that someone has uncontrolled diabetes. Certain medications can be sweat out that way as well. So there are other components that can be in that eccrine sweat as well. All right, like I said, that elimination of wastes. All right, questions on that? All right, excellent. That brings us to our second type of sudoriferous glands. That second type of sudoriferous glands are what are known as epicrine sweat glands. These are the ones we talked about with Michael Jordan, where again, they thought these used a epicrine mode of secretion, which is why they called them epicrine glands, but we learned that is wrong. So again, remember they are epicrine by name only. Right? Just because they're named epicrine doesn't mean it doesn't mean that they actually use the epicrine mode of secretion. Again, it's a bad choice of name, but just to remember that it is the name. Right? Just because someone theoretically was called Baron doesn't mean they get to, you know, rule the landscape over serfs or something like that. Again, it's just by name. All right. Now these, they're much, much fewer than the merocrine, or pardon me, the eccrine. Only about 2,000 on the surface of the skin. And these are actually associated with hair. However, not just any hair. These are the ones that are associated with secondary hairs. What do I mean when I say secondary hairs? What is a secondary hair? Again, we're going to get through this lecture a lot faster if you guys respond. Excellent, perfect. Hairs that are grown after puberty, hairs that are not present at birth, right? I was not actually, believe it or not born with a beard, right? As was mentioned, typically puberty occurs, the massive increase of estrogens and androgens that are produced by the male and female bodies cause the body to mature sexually. And part of that sexual maturity is the formation of secondary hairs. And those secondary hairs form in areas like the axillary region, like the pubic region, like the buccal region, the thoracic region. And associated with these hairs are these epicrine sweat glands. These epicrine sweat glands produce a more organic sweat. This sweat isn't associated with temperature regulation. Instead, like I said, it has a, what we typically think of, again, when first produced, as a more organic, a more musty, typically pleasing type of sensation. Now, again, if you've worked out at the gym, you throw your gym shirt into your gym bag and you leave it in the back of your car for 3 days. When you open that bag 3 days later, pleasant isn't the term that comes to mind. But remember, that smell we associate with body odor is actually due to bacteria feeding off of these epicrine secretions. And it is the bacterial releases that cause that gross aroma when you open that bag, or what we typically associate with body odor. All right, this is also why your 5-year-old can run around the house for hours and still smell like a rose. Whereas I barely want to go into my 15-year-old's room anymore because, right, it reeks from just all of the smell of all of her, everything that's going on in there. So again, we have these that form and become active upon puberty. All right. And so again, these are associated in those areas and only associated with those secondary hairs: axillary, anogenital, areolar, buccal region. I think it says beard region, but buccal region, right? Those areas of the body. Now notice when we look at these, where's my picture? Did I pass it already? Ah, perfect. No, I didn't. Wait, hold on. That's the one I wanted. Where did I keep? Bingo. Excellent. Notice when we look at this one histologically, comparing this to the previous one, notice that the size of the wall of the gland is much smaller in relation to the size of the lumen. This one has a much larger lumen. This is still a coiled tubular gland, but it has these massive lumens where it's producing a thicker, more organic secretion, right? That's why they thought it used the epicrine mode of secretion, because it was a more organic secretion. It just turns out that it uses merocrine. So the way you're going to tell these apart histologically is by the size of that lumen, right? If we go back and compare the two real quickly, the size of the wall to the size of the lumen here, small lumen, eccrine, large lumen, epicrine. That's how you're going to distinguish them and tell them apart. And notice they've done this here. And of course, notice the nice thing about this illustration is here, this one is feeding into a hair follicle, whereas this one just comes up to the surface of the skin. So that's another dead giveaway that this one here would be an epicrine gland. All right. Questions on that? All right, excellent. There are two more sudoriferous sweat glands. You need to know them, where they were found. You obviously need to know their mode of secretion and their structural classification, but you already know those because you know that they have to be simple coiled tubular glands. They have to use the merocrine mode of secretion. But I'm not going to hold you for them histologically. I'm not going to show you a microscope slide of these because they're too similar to the epicrine, and I find that to be confusing. But you do need to know what they are. The first one are what are known as ceruminous glands. And again, I'm showing this to you here just so you can see the similarity. You would not get a slide like this on the exam. If you see a slide that looks like this with the large lumen of this simple coiled tubular, your answer to that is going to be epicrine because that is what you're responsible for histologically. But you can clearly see here how it looks like that epicrine gland with a very thin wall and a very large lumen. However, while it looks like an epicrine gland, this is actually a ceruminous gland. Anyone know where ceruminous glands might be located? Anyone want to hazard a guess?
In the lumen?
There is a lumen associated with them where they release, and if you cheat, like you should be doing, and look at the picture, you see this term here. Anybody know what that term there refers to? Ears, absolutely, the ear hole. The location of your ceruminous glands are your ear hole. And guess what ceruminous glands make?
Serum, earwax.
Correct. What we commonly refer to as earwax, absolutely. What we commonly refer to as earwax, but you are absolutely correct. Ceruminous glands produce cerumen. Cerumen is earwax. What is the function of that earwax?
Protection
Protection from what?
from outside things like dirt and bugs or.
You're absolutely correct. Cerumen captures dust and debris because, again, your ear hole is an opening to the inside of your body, so that earwax captures dust and debris. But you're also right. It actually has a minor insect repellent capacity to it. Now, I'm not saying that the next time you go camping, you should just rub earwax on your body and you're not going to get bitten by bugs, but it does help to deter bugs from coming into your ears. So it does have a minor insect repellent capacity and it captures dust and debris. Absolutely. Excellent. The fourth and final type of sudoriferous gland is the mammary gland. And of course, where are the mammary glands located?
Your chest.
Yeah, your chest. Again, this is anatomy and physiology class. You're allowed to say breast. Absolutely, they're found in your breasts. And again, what is their function? But they produce milk. Exactly, they produce milk. Now, as I mentioned, one of the interesting things about them is there are two different kind of types of milk that the breast produces. The early milk, the fore milk, which is more watery, more sugary based, carbohydrate based, and the hind milk, which is more fat and protein rich. So it is thought that there may be different types of mammary glands. And as I mentioned, some of those they believe may use the epicrine mode of secretion, but we have not confirmed that information yet. So for right now, we'll stick with the company line. Mammary glands are specialized sudoriferous glands. So they are simple coiled tubes and they use the merocrine mode of secretion. All right. So there you go. Those are the 5 specific types of glands you are responsible for in the skin. Questions on that? All right, excellent. From there, we can then switch gears and talk about a hair. So any questions on glands before we move on? All right, so let me tell you a story. Here's the story. In this story, there is a single layer of cells. These cells divide very rapidly, and as they divide very rapidly, they push the cells above them up and they extend outward. As they extend outward, they fill with keratin and they die, becoming hardened, fused together cells. And in fact, down here at the base, there are actually some special cells that can produce a special pigment called melanin that fills these cells and gives them coloration. What did I just describe?
Melanocytes.
No, it sounds like I described the keratinocytes, and it sounds like I just described the epidermis and what happens in the epidermis. Isn't that basically what happens in the epidermis? One cell layers divide rapidly. Those keratinocytes produce keratin, fill up and die, harden and fuse together. There are melanocytes in here that provide pigmentation to it, causing it to grow. Yeah, that's exactly what happens in the epidermis, and it is exactly what happens in the hair as well. Hair grow in almost the identical fashion as skin, and in fact, it is those same keratinocytes and same melanocytes that are forming those cells. All right. Now, when identifying the anatomy of the hair, there are a couple things we need to take into consideration. The first, and again, remember, vocabulary is important. There are 3 regions to a hair. All right. Those 3 regions to the hair. Great question. Yes. So, Ryan, absolutely. In this case, the melanin that is produced in the hair does tend to stay in the hair longer so that it has that color further as it extends out till the end. However, what's also interesting about the melanocytes in the hairs is whereas the melanocytes in the skin typically continue to produce melanin for your entire life, for many people, and again, this tends to be genetically determined, in the hairs, for whatever reason, these melanocytes actually will go inactive and stop producing melanin. And when they stop producing melanin, what happens to the hair?
Becomes gray.
Yeah, it becomes gray or white, absolutely. So this gray or white hair we get is because the melanocytes associated with those hairs actually stop growing. They stop producing their melanin, and the hair color is now absent. All right, so the way a hair grows is very, very similar to what we see in the skin. We're producing keratin. That keratin hardens and dies. The cells harden and die. These keratinocytes harden and die, fuse together, and form a hair. There is one, so there are 3 regions to the hair. Let's talk about that first. The first region is actually this region down here where the hair actually grows. This region is called the bulb. The bulb contains the matrix, and that matrix is that single layer of keratinocytes that are dividing rapidly to be able to produce a new or continue to grow this hair. There was a question asked earlier about waxing. When we shave, if you think about when we shave, basically we're just cutting the hair off right here at the top of the skin. All right, and so then as it continues to grow, right, by typically 5 o'clock that day, some hair is already starting to poke out. So one thing that people will do is wax. Right? Waxing basically tears the hair out, and typically where the hair tears is somewhere about halfway down the hair shaft. And so down here in this region of the hair is where it breaks. and so you pull out a large portion of the hair, and so when you wax, it takes a lot longer for the hair to grow back and be able to come out. Sometimes when you wax, you pull the entire hair out. But as long as there's a little bit of that matrix left, the hair will grow back. The only way to get the hair to not grow back is to destroy the matrix. Now, back in ancient times, and by ancient times, I mean the, I don't know, 80s. In the 80s, it was suddenly decided that women having hair was gross. And so one of the big, huge things was people wanted to get rid of their hair. And so one of the processes that they would do is a process they called electrolysis. In electrolysis, basically what they would do is they would take a needle, they would insert that needle down into the hair shaft and basically produce a little bit of an electrical current. And that electrical current would destroy the matrix of that hair so that that hair would never grow back. The problem is they had to do it one hair at a time. So if you, for instance, didn't like how much hair you had on your forearm, it could take weeks of electrolysis sessions to get all of that hair removed because they basically had to do it one hair at a time. All right, as we've talked about before, the most important thing in life is apparently to be beautiful. So they have modernized this process, right? They don't have to go one hair at a time anymore. How do they destroy the hair matrices now?
Lasers?
Laser, absolutely. Instead, they focus a laser down at the matrix of these cells, destroying and ablating those matrices so that the hair will not grow back anymore. Right? So that region is the bulb. The rest of the hair is divided into two regions. Those two regions are the shaft and the root. Now notice your book here has put a line right here emphasizing where that dividing point is. Is it because it's halfway? No, it's a little more complicated than that. We have not yet talked about the layers of the hair, but we're about to do that in a second. But as many of you probably know, the outer layer of the cell, the outer layer of a hair is a very thick, very dense, very actually rough structure called the cuticle. This cuticle is the outer surface of the hair, and it provides a very strong protection to the hair, helping to maintain the integrity of the hair. One of the things that can happen is that if the integrity of the cuticle is damaged, then what can actually happen is the hair can lose its integrity and actually can start to come apart. And what do we call that when that occurs? Right?
Split ends.
Exactly, right? If you're like me, right, when you only have 4 hairs on your head, they all matter. So I get to my, my hair salon at least twice a week to make sure that they cut all those split ends so that I keep my beautiful coif that I have, right? So absolutely, that cuticle maintains the integrity, and if it breaks, you have those split ends. So the actual physical dividing point between the shaft and the root involve the cuticle. The root is considered the part where the cuticle is incomplete, where it is forming. Root, sorry, not rot. The root is where the cuticle is incomplete, where it is forming. Once the cuticle is complete, that part is considered the shaft. So the shaft has a full, complete cuticle. Now, while your textbook has drawn the line here, even in the illustration that they've given us, can we see where the cuticle is complete and where the cuticle is incomplete? It's not a trick question.
Not
Can you guys
really a
actually see
big
that?
difference.
No, there's not a difference here as well. So what we are going to do is we are going to simplify the rule for us. Basically, what I'm going to do in this class is we're going to make it easy. If the hair is extending out of the skin, for this part where it's extending out of the skin, does the cuticle have to be complete if it is sticking out from the skin?
Yes.
Yeah, absolutely. So the portion that sticks out, we are going to refer to as the shaft. And then, so for us, everything, and let me go ahead and erase these two here. Everything then that is below the surface of the skin, all of this part right here, we will call the root. We're cheating a little bit. We're simplifying it a little bit, but I think it is a necessary evil and will help to make sense. So basically, all the hair except the bulb that is inside the skin, we will consider the root. All the hair that is extending out, we will consider the shaft. And of course, the large part at the base is the matrix. All right, are we comfortable with that definition? Again, we're cheating a little bit, but we're simplifying it to make it easier to understand. All right, so 3 regions to our hair. The 3 regions to the hair are the shaft, where we have that hard keratin outer layer complete. We have that cuticle complete on the outer surface. The root, where the keratin is forming. And we have the bulb, where the matrix is located. Now, I want you to think about one other thing as well, and I want you to notice this as we look down here at the drawing at the bottom. Again, the matrix is the single layer of cells, and as we talked about, they're keratinocytes, and they're dividing and they're moving up, and that means they're epithelial cells. All right? And if they're epithelial cells, that means they're avascular. But this matrix needs to be able to divide. So it needs a lot of oxygen. It needs a lot of nutrients. So notice there is a finger-like extension of the dermis with a large capillary in it that penetrates up like a finger-like extension into the bulb of the hair. And guess what we call that finger-like extension of the dermis? We call it a papilla. So notice here we have that hair papilla extending into the bulb of the hair where the matrix is going to be, and that matrix will also be where the melanocytes are located. All right? Questions on that? So those are the regions of the hair contained within our follicle. But the other thing we can do is we can cut the hair and the follicle in a cross section. And if we cut the hair and the follicle in a cross section, we can see the layers of the hair. And there are 3 layers to the hair. Now, I don't know if we've done this in this class yet, but it is a common organization that we are going to see a lot in this class. Often in this class, we are going to see one thing inside of another. We are going to see these structures that have a chewy nougat center and a candy-coated shell, an inside and an outside. The hair is this way. Our adrenal gland is this way. Our kidney is this way. We'll see it in the bones. We'll see this a lot of ways. This is an organization we're going to see a lot, and these are terms we're going to use a lot as well. When we have this type of organization, the inner region is known as the medulla. So the medulla is the inner region, and the outer region is known as the cortex. So in our hair, we are going to have the innermost region, which is the medulla. That's the part here that is light tan in color. And the dark brown region, the outer part, is the cortex. All right. But remember, we said the hair has 3 layers, and that's because the 3rd layer that forms in the shaft is when we have that very thick, very, as I mentioned, coarse outer layer that is the cuticle. So this big, thick outer layer is the cuticle of the hair. Now again, it's not present at the bulb. It forms during the root, and it is complete in the shaft. Now, when you pull a hair out of your head and look at it, it looks all pretty and smooth. And the reason it looks all pretty and smooth is because of that cuticle. However, if we were to actually look at that cuticle under the microscope, this is actually what the cuticle looks like. Notice the cuticle looks like shingles on a roof, overlapping layers of thick, hard, keratinized cells that are providing that strength, providing that integrity to the hair. So here, when you look at it in an electron microscope, what looks like a smooth, continuous layer of hair is actually this very thick, very coarse, very hard and protective outer surface that is the cuticle. And clearly we can see this is where it's coming out of the skin. So obviously this is all the shaft with that fully intact cuticle on its surface. All right. Questions on that? All right, excellent. Now, notice those are the 3 layers. And let's write that again. There are, stop it, 3 layers to the hair. But the hair needs to be protected. Again, the skin is our protection from the outside world. So we can't have hairs just sticking randomly out through it. Those would be a bunch of holes that harmful things could get inside. So that hair is surrounded by a structure known as the hair follicle. And that is what is represented by this structure here. That is the hair follicle. And it turns out the hair follicle is comprised of 3.5 layers. Now I say 3.5 because it's really kind of 4 layers or 3 layers. Well, I'll explain why 3.5 in just a minute. All right, I have a pretty picture, and here is that pretty picture. And let's take a look at this. What we have here is a cross section through the hair and the follicle. So notice as we look at it, we have the medulla right here. We have the cortex right here, and we have that hardened cuticle right there. So notice all of this, and I'll see if I can actually, I'm going to draw around it because I'm horrible with these circles. This right here, this line represents the hair. And so notice this hair is surrounded by 1, 2, 3.5 layers. This first layer of the follicle, and I'll put a nice little 1 right here to represent this. Oops, no, I wanted to do that. And then put that there. Represented here in orange is, as you can see, what is referred to as the internal epithelial root sheath. Internal epithelial root sheath. It's internal because it is the one that is closest to the hair. Next to it, here too, is the external epithelial root sheath. These are both very cell-dense layers that are going to help to surround the hair and help to maintain the boundary of the hair. In fact, if we cheat and go back to the previous picture, notice the external epithelial root sheath is really an extension of the epidermis. The epidermis comes down and expands all the way around the hair to maintain that barrier with the outside world. So really, if you think about it, that external epithelial root sheath is really a continuation of the epidermis. However, we do have this issue. Both 1 and 2 are epithelial tissues. And we know 2 things. We know a lot of things about epithelial tissues, but 2 of the important things we know about them is that they're avascular, meaning that we need to provide blood and nutrients and oxygen for it somehow. But the other thing we know about epithelial tissues is what is on the base, or let's be more specific, the basal surface of all epithelial tissues.
The basement membrane?
A basement membrane. Excellent. Perfect. And remember how I told you that if you use your glue properly, you'll never see the basement membrane? but I also told you there's one exception. This is the one exception. In some instances, and again, you're not going to see it on every follicle, but in many of the follicles, you will actually see a little bit of a clear layer here between the epidermis and the dermis. And again, I know with my drawing skills, I'm not doing a great job of drawing this, but this layer here is the basement membrane of the epithelial tissue. But it can actually be seen on some of our follicles. And so this basement membrane here in this location only is referred to as the glassy membrane. Oops. The glassy membrane. So the glassy membrane is basically the basement membrane of our hair follicle. And we've got some decent pictures where we will actually be able to see them. Of course, that glassy membrane holds it to its tissue that's around it. And so then here is layer 4. We'll call it layer 4 just because we'll call the glassy membrane layer 3. And this outermost layer is what we call the dermal or connective tissue root sheath. All right, so again, with our, let's go ahead and clear all of this. We have that internal and external epithelial root sheath, the glassy membrane, and then our dermal or connective tissue root sheath. So think of it this way. If we were cutting through a hair follicle from the outside, we'd go first through the connective tissue root sheath, glassy membrane, external epithelial root sheath, internal epithelial root sheath, cuticle, cortex, medulla. Those are the layers of the hair and the layers of the follicle. All right, questions on that? All right, excellent. The hairs are important. The follicle is important, but there are also some structures that are associated with the hair as well. Some of them, as we've talked about, are the sebaceous glands and the apocrine glands that would be associated with secondary hairs. And notice one other thing. Again, we are talking about hair as a singular thing because remember, in this class, the sky is blue. The hair on your head, the hair on your chest, the hair on your legs, your pubic hair, your eyelids, your eyebrows, all of these things, your eyelashes, all of these things are hairs. But do they all grow at the same rate? Do they all have the same characteristics to them?
No.
No. So there are different types of hairs that have different growing rates and different characteristics and things like that to them as well. We are simplifying basic rules for hair. So we're kind of lumping all the hairs into just one thing. So I do appreciate that. Again, as we've talked about, every single topic in this class is more complicated than we're talking about. We are simplifying things a little bit. But for hairs, there are some other important structures. And the first are the erector pili muscles. Erector pili muscles are little bands of muscle that help to move the hairs. These hairs, and again, the illustration doesn't do the best job of showing this, so let's go ahead and draw it. Most hairs are in the skin at an angle. And you can actually see that. If you look at your forearm, you'll see the forearm, the hairs, for the most part, are laying down on the surface of the skin. But what these erector pili muscles do is they connect from the follicle to the underside of the epidermis. And when these muscles contract, that pulls on the hair. And as it pulls on the hair, it causes the hair to stand up. When it stands up, it also causes this muscle to bunch together, because as the muscle shortens, it bunches up and gets larger, and causes that condition we refer to as goosebumps. So your hairs stand up on end and you get those goosebumps as a result of it, and that is because of the erector pili muscles. Why? Why do these hairs stand up on end? What's the purpose of that?
Is it sensory? Or
Possibly. Excellent. One is definitely to play a role in keeping the heat close to our body, right? If you think about when you get goosebumps, you get goosebumps when you're cold. That's one of the times that you do it. And again, by quick show of hands, yes or no, or you can type yes or no, or you can just say it out loud. Has anybody here scuba dived before? No, nobody. Okay, well, none of you apparently have scuba dived. Have any of you seen scuba divers before? How about a James Bond movie where he's like in the scuba suit and he gets out when he gets to another location, takes it off, and he's got his perfectly pressed tuxedo underneath? Is that actually the function of a scuba suit? When you use that scuba suit, is that scuba suit to keep you dry? No, excellent. It's not. In fact, when you put on that scuba suit, what you actually do is you actually put water inside the scuba suit. The reason for the scuba suit is actually to help you with your body heat. What happens is when you're exposed to water, water transmits heat much easier than air does. Right? Again, this is not something that we think about every day, but I know it's something you're aware of because you can sit in your household for hours, 5 straight hours in your house with the house at a nice, comfortable 75 degrees, and you feel fine. If you sat outside in your pool where the water was 75 degrees and you stayed in that pool at 75 degrees for an hour, would you be comfortable after that hour? No, you'd be cold because that water steals your heat. And so the point of that scuba suit is that when you're swimming, water comes by, rubs across your body, it steals your heat, and then the current carries that water away. But with that scuba suit, you keep the water close to your body. And by keeping the water close to your body, it still steals your heat, but you're keeping that warm water close to your body, and it helps you to maintain your heat more, right? It's the same concept with those Siberian Husky dogs. Right? You're up there doing the Iditarod instead of scuba diving. And what happens is you have those dogs and they have that ability to make their hair stand up on end. That hair standing up on end captures air around it, keeps the air close so that as the wind blows by, it captures and keeps some of their body heat and they stay warm. So if we go back to homeostasis from day 2, as we talked about, you stepped out onto your front porch naked to pick up the newspaper, right? And the door locked behind you. And there you are shivering in the morning, but luckily your hair is going to stand up on end and suddenly you're not cold anymore, right? Is that what happens? If you're not sure, you can try it tomorrow morning. Step outside naked and see if your hair standing up on end keeps you warm. Is it going to keep you warm? No, of course not, right? We don't have as much hair as that Siberian Husky does, but the biological evolutionary process is still there, right? Maybe if you're as hairy as Robin Williams, or at least that's the joke I used to make. Now he's dead, he killed himself, so it's not as funny to make that joke, but I can't think of anybody else hairy, so you get the idea. All right, if you had a lot of hair, then maybe it would work, but otherwise, no. All right? I noticed someone else said nervous. The other time your hair stands up on end is when you're scared. And again, think of terms in your cat. When your cat gets scared, what does it do? It hunches up its back, it makes its hair stand up on end so that it looks bigger and more intimidating, right? Same thing for you. You're walking down the street with your loved one, someone tries to rob you, but your hair stands up on end, they get intimidated by how big and scary you are, and they run off. All right, again, probably doesn't work that way, but those evolutionary biological processes are still in place. When we're scared, when we are cold, our hairs stand up on end. They're not very functional, but they still are there to try to help us with those processes. Now, it doesn't mean they're useless. If you notice closely as you look, I pointed at the screen like you guys can see my finger. If you notice here, that erector pili muscle happens to be right up against that sebaceous gland. So what happens is now contractions of this actually help to express our sebaceous gland. So our erector pili muscles do still serve an important function. They help in the expression of our sebum by contracting. And one last thing, we said this is muscle, but what type of muscle are these erector pili muscles? We know muscle comes in 3 flavors. Which flavor is the erector pili muscle? What type of muscle is it? Well, is skeletal? Can you voluntarily control it? Rise, and they all stand up on end? No. Perfect. Smooth muscle. Excellent. This is smooth muscle. We involuntarily control it. So this is something we cannot voluntarily control. All right. So it is smooth muscle. Excellent. So that's one structure associated with our hairs. As we also mentioned, the sebaceous glands are associated with it. The other structure that we haven't talked about yet, but we need to, is what is known as the hair root plexus. Plexus is a fancy word we will talk about a lot in this class, and plexus means network, an elaborate network. As it turns out, there is an elaborate network of nerves that wrap around the follicle of our hair. So, as you're sitting here talking to me in class, if a fly were to land on the surface of your skin, would you be aware of it? Yeah, exactly. These provide sensation. If someone were to move your hair, if someone were to grab your hair and pull it out on your arm, would you be aware of all of those things happening? Absolutely. And so that hair root plexus gives us that sensation of the movement of our hair. So we have those sensory structures associated with the hair as well. All right. Questions on that? All right, excellent. One more. The nair. Stop me if you've heard this story before. We have a single layer of keratinocytes that divide rapidly. As they divide rapidly, they push the old cells up. These cells fill with keratin. They harden, they fuse together, and they die till they reach the end where they're chewed off or cut or filed or whatever, right? The same way the skin grows, the same way, or the epidermis grows, the same way our hair grows is the exact same way that our nail grows as well. Our nail grows from a single layer of cells known as the nail matrix. And as that nail matrix grows, it produces new keratinocytes. When those keratinocytes are still under the skin and not fully keratinized, they form the nail root. As they completely solidify and keratinize, they then form the body of the nail, which then comes out to what we call the free edge, where, like I said, is where you cut, where you file, where you chew. All right. This matrix determines the growth of your nail. So if, like me, in middle school, I was carrying, because I grew up back east, I was carrying a load of logs up the stairs from the basement. Back east, they have things called basements. And I tripped and fell, and a log smashed my thumb. Damaged my thumb pretty badly, and actually my nail fell off as a result of the injury. However, my nail matrix was intact, and so the nail grew back. Unlike my wife's best friend, who has twins. And she was getting her 2-year-old twins into the car, and as she was getting her 2-year-old twins into the car and closing her door, one of her twin daughters got her pinky stuck in the door as it was closed and lost her nail and destroyed her nail matrix. And so guess what she doesn't have anymore? She no longer has a pinky nail. By destroying that matrix, her pinky nail never grew back. But the good news is now she can easily tell the two kids apart. All right. So we have that matrix where we're constantly growing our nail as a result of that. There are a couple unique characteristics, features that we need to identify. The first, notice because of the thickness of the matrix. Remember, the matrix is epithelial cells, and epithelial cells are avascular. There is no blood supply to them. As you look at your nail, and again, this is one of those silly things that I have to make sure I mention now because it happened. What you should be able to do is squeeze your finger, and as you squeeze your finger, what you'll see is that it turns white. And then as blood comes back to that area, it turns pink. Now, this happened in the classroom, so this wasn't something that happened recently, but several years ago, I had a student who panicked because as she squeezed her thumb and looked at it, she didn't see a color change because she was wearing nail polish. So if you're wearing nail polish, this won't work. But as long as you're not wearing colored nail polish, you should be able to squeeze your thumb or any finger, see the blood go out of it, have it turn white, and then see the blood flow back into the area. However, if you notice at the base of the finger, where that matrix is larger underneath, because it is thicker, because there's less blood vessels in that area, we have a slightly light colored half moon shaped region, and that half moon shaped region is what is known as the lunula. All right. There are two other important features. I'm going to go ahead and clear my drawing here so that we can identify these, and I'll actually switch the color. Obviously, these is attached to the base, but there are two thickened portions of the skin, because again, this is something that is extending out of the skin, so we need protection. There is what we commonly refer to as the nail bed. However, the nail bed is not an appropriate anatomical term, so we will use the appropriate anatomical term of the hyponychium. That hyponychium is what anchors the body of the nail to the distal portion of the finger, right? Any good spy movie worth its salt, at some point, our protagonist is threatened with bamboo shoots being stuck underneath their nail bed, right? Because this is a very sensitive region. That would be a very painful sensation that would occur, and that's that attachment point of the eponychium. The other important feature is a thickened fold of skin at the base of the nail body, what is known commonly as the cuticle, but again, you will use the appropriate anatomical terms. If you use the term nail bed and cuticle, you will not even get partial credit for that, 0 credit. I want appropriate anatomical terms. The eponychium, but the eponychium is what people commonly refer to as the cuticle. You know, 10, 15 years ago, one of the things that would happen when you went to get your nails done is when you went to the nail salon, they would actually cut off the cuticle, right? Because if you cut off the cuticle, that exposes more of the nail, makes the nail look longer and more beautiful. And so it was something that was very commonly done. Well, not surprisingly, if you cut off the cuticle, you open up the nail matrix to the outside world, and it dramatically increased the number of infections that occurred as a result of that. So now typically what happens is they try to just push back the cuticle so that it doesn't expand out so far, but typically it is not cut the same way that it was before to expose that, because again, it dramatically increases the likelihood of a nail infection occurring as a result of that. All right, so there you go. That is the basic anatomy of our nail. Questions on that? Excellent. We did all that. Perfect. Excellent. The good news is with that, we are done with the anatomy. The other good news is we've talked a fair amount about the physiology as we have gone through this as well. So what we're going to do now is, again, looking at the time, we're going to take our second break. When we come back from the break, we are going to talk physiology. A lot of this physiology is going to be review or synthesizing information we've already talked about. So there actually won't be too much new information. We'll be expanding on some of the things we've talked about, but there won't be too much new information because we've talked about a lot of these things as we work through the anatomy. So we're getting close to the end. Let's go ahead. I see 10:30. Let's come back at 10:45, and at 10:45, we will talk about our physiology. So we will restart at 10:45, and I will start the recording at that point. So any questions before we take our break? All right, excellent. Perfect. So again, to finish off the anatomy, these images of these models are in your modules now on Canvas, Skin Model 1 and Skin Model 2. What's nice about them is not only do they show the skin anatomy, but they also show 3 distinct regions. Obviously, we can see the clear division here between our thick skin and our thin skin that is over here. That is what is being represented by this division. Notice here, and again, you'll be able to increase the magnification and look at this. This yellow region right here is the stratum lucidum that thick skin has, that the thin skin doesn't. A much thicker stratum corneum. And notice the only glands that are present are those thin, small sudoriferous glands, the eccrine sweat glands. However, over here, where we have hairs in the thin skin, we have a thinner stratum corneum, no stratum lucidum, but we can see the other layers. We can see our sebaceous glands that we have here connected to the hair follicles, as well as the eccrine sweat glands, which we see everywhere. And notice also connecting to this hair, we have a much larger simple coiled tubular gland with a larger lumen representing the epicrine sweat gland. So this is actually the axillary region, whereas this over here would be like, for instance, your scalp or your forearm. So we see that anatomy of that here. We see one of those Pacinian corpuscles. We see our tactile corpuscles. We see some of the things. We see a lot of the anatomy, the capillaries and the papillary region, and all that kind of stuff that we've talked about. Here is the second skin model. Notice it is also divided into 3 regions. Thick skin over here. And again, now we can more clearly see the 5 distinct layers and the 4 distinct layers of the epidermis on this. Notice we have a free nerve ending as well as a tactile corpuscle, and the Pacinian corpuscles are lamellated corpuscles. That pseudorefers gland that is specifically eccrine. Here we have our epicrine with a larger lumen, and it's connected directly to the follicle. Here we see our sebaceous gland. But what I like about this model in particular, and whenever you hear me say words like that, that I like something, that almost guarantees it's going to be on the exam, is if you look closely at this one does a great job of showing us the different layers associated with the hair and with the follicle. So last time we went from outside in, this time let's go from inside out. The innermost layer is, of course, the medulla. That would lead to the cortex. That would lead to the cuticle. So those are the 3 layers of the hair. Then we have the internal epithelial root sheath, the external epithelial root sheath, the glassy membrane, and the dermal or connective tissue root sheath. We even have a little bit of an erector pili muscle that we can see here as well. Right, so again, these do a great job of showing these things really nicely. You're responsible for the anatomy of the hair on models, on charts, on histology slides, all of those ways. But I want you to be able to study these models, so I've made these 2 pictures available for you because it's definitely stuff that could be on the exam. All right, questions on that?
Yeah, quick question. Sorry, for some weird reason, I got disconnected before we came back during the break. How did we get to that model you just showed previously?
So so if you look at our modules, remember there are the different sections, like for instance, where the lab handouts are and the lecture slides and those are. Remember the last section are images from lecture. I have put them in the images from lecture. So you will find them there. I literally just posted them on there, so they should be there now. You should see those 2 files there now.
Gotcha.
All right?
Okay.
All right, excellent. So last little bit of business we need to take care of in the last hour and a half of class is to talk about the physiology of the skin. And like I said, the good news is that there isn't as much new information. We're synthesizing information. We may be expanding on it a little bit, but most of this is stuff that we have already talked about. All right? Like for example, we have already talked about coloration of the skin. When we talked about coloration of the skin, obviously we talked about the main pigment that we think of in relationship to the skin. And again, that main one is melanin. We talked about melanin, how everybody has basically the same number of melanocytes, but how they're different is in the amounts of melanin produced and the colors, the different types of melanin that are produced. Melanin, as we talked about, have over 4 genes that are involved in it. And because of those variations, you can have anything from a more yellowish or reddish pigment to brown and black. And again, you've done your biointeractive where we've talked a lot about melanin in that as well. However, there are 2 other pigments that can add to skin color. One of those is keratin. Keratin is actually a precursor for vitamin A. So where do we get keratin from? It's not your question. Where do you think we get keratin from?
Protein.
Well, you got the right idea. We get it from our diet. Unlike melanin, which is made within our skin, we actually get keratin from our diet. And what types of things do you think might have keratin in it?
Carrots.
Carrots. Carrots and other orange vegetables. Carotene is a yellow-orange pigment to it. We typically get it from our diet, and it is stored typically in the more superficial layers of the skin. Remember, with melanin, as we talked about, this is typically stored just in the deep layers. And again, if you think about it, that makes sense. Our deep layers are where our keratinocytes still have a nucleus. The dead cells on top don't have a nucleus. So do we really have to worry about protecting them from UV radiation? No, it doesn't have any DNA that we have to worry about it mutating. But keratin is typically stored in the more superficial layers and also the deep fatty layers of the hypodermis below the skin as well. It typically doesn't play a huge role in our coloration of our skin, but there are instances where it can become more extreme. You see this, people like Jack LaLanne or other people like that who become very large proponents of juicing and juicers. Typically people who juice a lot of vegetables tend to get a lot more keratin into their diet, and they typically take on a darker, more orangish coloration, bronze coloration to their skin. And I actually had a student, 7, 8 years ago now, who one summer, her son decided he was going to eat nothing but carrots. And after doing that for 3 weeks, he actually got a nice orangish coloration to his skin, which he took a picture of and shared with us in the class. So that was very entertaining that way. Of course, the last pigment is something that we've talked about, but you probably haven't thought of it in terms of the pigmentation of the skin, but you're aware of it. Because as I mentioned, again, it's easier when we're in the classroom. In the classroom, you guys would have to stand up and give your presentations. And if you were standing up in front of the class giving your presentation and your pants fell down, would there be a change in the coloration to your skin, especially maybe the skin of your face? Yeah, absolutely. Yes. And what would cause that change in the coloration of your skin? What color would your skin become?
Is that like the blood rushing to
Red.
your face?
Yeah, become red because of the blood, absolutely, because of the hemoglobin. Remember, hemoglobin binds oxygen, giving it that reddish coloration, and so that red pigmentation to the skin is due to the oxygen in our red blood cells. Now, again, skin color is a huge social issue right now, but from a clinical standpoint, coloration of the skin is important because it tells us a lot about the condition of the body. We can learn a lot about the condition of the body by looking at the coloration of the skin. One example of that is if the skin were to become cyanotic. What color is the skin if you are cyanotic? Right, it's going to have a bluish coloration to it. Absolutely. And that bluish coloration occurs typically from a decrease in oxygen in the blood, or at least the decrease in oxygen-rich blood in the skin. There are two possible causes for that. One, when you get cold, the blood vessels of our skin constrict, drawing the blood away from the surface of the body. So we have less blood close to the surface, and the skin takes on a more whitish or a more bluish appearance. But the other reason is a lack of oxygen, right? So again, if you've got a pulmonary embolism, or someone's got a plastic bag over your head or something like that, your skin's going to take on a bluish coloration because of that lack of oxygen getting to your tissues of your skin. Conversely, what color is your skin if you are erythemic? Red coloration, absolutely. The red coloration here is from a congestion of blood in the skin. When you're embarrassed, increased blood flows to the skin. Or injury, right? You walk up to that cute gentleman at a bar, use some cheesy pickup line, and he slaps you across the face. Okay, probably not likely to happen, but all right, maybe the other way around. You walk up to some girl, give her a cheesy pickup line, and she slaps you across the face, and you have this nice big red handprint on your face from, again, that injury and the congestion of blood. Or with a bright sunny weekend, maybe you're going to go outside and with your loved ones and study out in the backyard, and you get a little bit of a sunburn. That redness that comes from the sunburn is from a congestion of blood in the skin. And if you don't believe me, then here's what I encourage you to do. The next time your loved ones get a sunburn, what I want you to do is take your hand, come up, and press it very firmly on their back. And when you do that, press it very firmly on your back and pull it away, what you will see is a white handprint on their back, because what you've actually done is push the blood out of that congested area. Of course, the blood will rush back into the area, so you can do it again. In fact, I encourage you to do it 3 or 4 times to them. Explain to them it's for science, and it will all be okay. All right? What color is the skin if it's jaundice? Right, why? Where does this jaundice yellowish coloration come from? What causes it? Anyone know? Close. It is associated with the liver and bile. You guys have the right idea. The liver does produce bile, but also part of what it produces the bile from is a special protein called bilirubin. Bilirubin is a protein that actually comes from the breakdown of red blood cells. Remember, as we talked about, red blood cells are not true cells. They don't have a nucleus. They're just big bags of hemoglobin. And so their lifespan is only about 100 to 120 days. You're constantly breaking down red blood cells and constantly making new ones. When we break down the red blood cells, that hemoglobin is broken down into a molecule called bilirubin. That bilirubin is used to make bile. And in part of that process, that bilirubin is broken down into 2 components. Stercobilin, which is a brown pigment. And that brown pigment, the stercobilin, actually helps to color your feces. It's released in from your digestive system. The other pigment it breaks down to is urobilin. Urobilin, oops, that's not an R. Urobilin is actually reabsorbed by the digestive system and released in your urine, and is the primary coloration of our urine. So the color of our feces and the color of our urine comes from the breakdown of the bilirubin, and this bilirubin is broken down in the liver. However, if there are problems with the liver, you've got some kind of liver disorder, or maybe you've been drinking vodka for breakfast, lunch, and dinner for 90 straight days, right? In that case, your liver becomes less functional, and it's not able to fully break down the bilirubin, and bilirubin accumulates in the blood, giving that yellowish greenish coloration to the skin. Now, where else is jaundice an issue?
Does it
Yeah, go ahead.
Professor, jaundice, does it appear in babies?
Exactly. That was the second thing I was going to talk about. Jaundice is also very common in newborns. In fact, a recent study shows that about 30% of newborn babies show some level of jaundice. It's very normal. It's very common. The reason for this is fetal blood actually has a lot more red blood cells in it than normal adult blood does. And one of the things that happens at birth is that a large number of those red blood cells die. The problem is baby's liver may not be fully functional yet. And if it's not fully functional yet, it can't keep up with all of this massive death of red blood cells. And so the baby gets jaundice as a result of it. It's fairly common for it to occur. Now, the good news is it's fairly common, and it tends to follow a fairly standard path. The problem is that if bilirubin gets too high, bilirubin can actually disrupt or interfere with neural development. And obviously, neural development is something that is super, super important. So what happens is if it gets too high or it doesn't come down rapidly enough, then they need to do something to intervene. And what is it typically that they do to intervene with babies that are jaundiced? What do you do with babies? What did grandma do with babies when you were jaundiced? Yeah, grandma would put them on the windowsill, right? Not quite like a flower pot, right? But a nice diffused curtain, and they would get that indirect sunlight, because you guys are absolutely right. UV radiation, as it turns out, breaks bilirubin down into a more easily digestible form. So that's why when you go to any NICU center, it's just, you know, rows and rows of windows with windowsills on it that they put the babies in. Okay, not quite, but you guys have the right idea. They have UV lights, they have UV blankets that they can put on them to help to more readily break down that bilirubin. Excellent. All right, one more example. What color does the skin become with carbon monoxide poisoning? Bright red or even almost a pinkish coloration, right? One of the interesting things, and I'm going to cheat and draw this really simple. Remember, as we talked about, hemoglobin is made up of 4 globular subunits. And at the center of each of those is our heme, and that heme has an iron at the center of it. That iron is what the oxygen binds to so that the hemoglobin can carry that oxygen. A single hemoglobin has 4 irons, so it can carry 4 oxygen. And a single red blood cell has about 250 million hemoglobin inside of it. So obviously a red blood cell has a massive carrying capacity for oxygen. And it's important because it's the only way your body gets the oxygen it needs. The problem with carbon monoxide is carbon monoxide binds more easily to the iron than oxygen does. It outcompetes it. And when it binds, it doesn't like to let go. So when it binds to our hemoglobin, right, oxygen can't bind to it. We can't get the oxygen to our body, and our tissues can be damaged, and especially tissues like our heart, tissues like our nervous tissue that are highly sensitive to oxygen levels can be permanently damaged by that carbon monoxide poison. So we all have to have those carbon monoxide sensors in our house. If that sensor goes out, or you start to see that your loved ones start to get very pink or bright red, grab them, get out of the house, get out of the into open air, and get help immediately for that. So these are all examples of how the color of someone's skin can tell us a lot about their overall condition. All right, questions on that? All right, excellent. Then let's talk about thermal regulation. One of the important functions we talked about the skin is the skin, because again, we were talking about the skin, helps us to regulate our body's temperature. Give me some examples of the way that our skin helps us to regulate our body temperature.
Sweat.
Excellent. By producing watery sweat, right? And what produces that watery sweat again? What gland produces watery sweat? Eccrine, excellent. Those eccrine glands, perfect. Eccrine glands, and again, that would be the specific type. What would the general type be? What would the general type of gland be?
The pseudophorosis.
There you go. Oops, did that wrong. Sudoriferous. Excellent. So the general type would be sudoriferous, but in this case, we want to be more specific. Eccrine sweat glands. Remember, they produce about a half a liter, but can produce up to 8 liters of sweat in a 24-hour period of time. This sweat produced from our eccrine sweat glands is what we call our sensible sweat, or let's say sensible perspiration, right? Now, again, I don't necessarily know what you were doing during your 15-minute breaks, but you probably weren't running around too much. If you looked at your forearm right now, do you see any sweat on your forearm?
No.
No, you don't. But guess what? You are actually perspirating from your forearm right now. It's actually what we call insensible. Again, sensible means to be able to perceive. We do have insensible sweat. That insensible sweat is actually the loss of interstitial fluid through the skin. Now, our epidermis is mostly waterproof. So do we lose a lot of moisture from our body this way? No, only about 500 milliliters during the course of the day. But as we talked about, if you have severe burns over long portions of your body, that affects the integrity of your skin. And one of the two main concerns we have for people who have major burns is infection, but also water loss, dehydration from that loss of that insensible sweat. We think of skin as being dry, but it is a tissue. There is interstitial fluid in there, and we do lose some of that during the course of the day. So yes, both sensible and insensible sweat is one of the ways, primarily sensible sweat. We do sweat insensibly, but this sensible sweat is one of the main ways we regulate our body temperature. Excellent. How else do we regulate our body temperature in the skin? True,
Is that
absolutely. Those goosebumps, or again, and what causes those goosebumps again?
cold?
Well, yeah, but it's the erector pili muscles. Oops.
Oh, sorry.
Those erector pili muscles make the hair stand up on end. Again, do they do a good job of helping us to regulate our body temperature? No, they don't do a very good job, but it is one of the processes that our skin does to try to help regulate body temperature. What else? These are all
Blood,
great, but exactly.
like blood vessels.
Absolutely. In fact, the primary way that our skin helps us to regulate body temperature, the primary way is actually with blood flow. All right? Think of it this way. If you were to get too hot, you get up and you run around the room 16 times. What happens to the blood vessels in your skin?
Increases?
Right, well, yeah, but more specifically, I think Nicole's got it. The key word is they dilate. Right, they're going to dilate as a result. There's dilate, and when they dilate the blood vessels, they bring more blood to the surface of the skin, and when they do that, we radiate off the heat. Right, so we bring that blood to the surface so we can radiate off that heat. Right, someone who's worked out really, really vigorously, you don't even have to touch their skin. You can just get close to them and you can feel the heat radiating off of their skin. Their skin is bright red as color. But what happens if you get too cold? What happens then?
They restrict, or
Yeah,
are the proper words?
exactly. We constrict. We constrict the blood vessels, and when we constrict the blood vessels, that takes the blood away from the body, away from the skin, away from the surface. And when it takes away from the surface, it helps us to store the heat. It keeps that warmth towards the core of our body, right? And that's good for short term, but it can become an issue if we're exposed to cold for long periods of time. If we're exposed to cold for a long period of time, then especially the smaller structures of our body, like our fingers, like our ears, like our nose, when that warm blood is pulled away for a long period of time, that tissue can actually be damaged by the cold. It's not getting as much oxygen as it needs, and that tissue can actually become necrotic and die. And what do we call that condition?
Frostbite?
Frostbite, exactly. So again, extreme constriction, extreme loss of that blood from prolonged exposure can actually cause that frostbite. So in this way, by changing our blood flow to our skin is a blood reservoir. While you're sitting here at rest, there's a large amount of blood just slowly chugging through your skin. And so moving that blood into and out of the surface of our skin helps us to regulate body temperature. Excellent, excellent, excellent. So again, we'll go through it again. Large supply of blood, it's that blood reservoir, changes in relation to temperature. When body temperature is high, we dilate, bringing more to the surface. When temperature is low, we constrict it, bringing it away from the surface. We talked about insensible and sensible perspiration. We talked about that. We talked about the hair standing up, goosebumps. What I'm thankful that we didn't talk about is shivering. Is shivering an important process for maintaining body temperature?
Yes.
Yes.
Absolutely. But when we shiver, we're using our skeletal muscle. And is that part of the integumentary system?
No.
No. And remember also, we talked about how our subcutaneous fat helps us to regulate our body temperature and provide insulation. But is that part of the skin? Is our subcutaneous fat a part of the skin?
Isn't that the skin?
It is not part of the skin.
It is not part of the skin. Remember, we said that our subcutaneous layer, that adipose layer, is associated. It loosely attaches the skin to the muscles, but remember, we said it's not a part of the skin. Remember, we also called it, it's subcutaneous. It's below the cutaneous membrane. It is not a part of the skin. All right? Questions on that? All right, excellent. Okay, perfect. Here we have the pretty picture from your textbook. What we can easily do here, let me move this out of the way. We've named them all, but I want to put them all together. I want to identify our tactile, our sensory structures. Let's do it this way. For each sensory structure, we need to say the type of information that we receive from it. We also want to identify the layer / sublayer where we would find it. So based on what we've talked about, and we have the pretty picture here to help us with that, can someone give me an example of a particular tactile sensory structure that we have identified so far in this class? Again, I know them. I'm doing this for you. Look at the picture if you can't figure it out. Excellent. So one of the things we talked about were the tactile corpuscles. Right. You can call them the Messner's corpuscles as well. Corpuscles as well. That is fine, but make sure you call them Messner's corpuscles and not just Messner's. Make sure you have Messner's corpuscles or tactile corpuscles. Excellent. What type of information? Oh, let's say, hold on, let's also do this. And if necessary, region. Excellent. All right. What type of tactile information did we say the tactile corpuscles gave us? Tactile cells is another type. We'll talk about those too, but you're absolutely right. Fine touch. Excellent. I like that. And we can write it that way. Fine touch. Or again, we said that fine discrimination. But fine touch is perfectly acceptable as well. Excellent. What layer or sublayer, and again, remember, we always want to be as precise as possible. What layer / sublayer do we find the tactile corpuscles in? Now, I'll give you a hint. It's the dermis, but we can be more specific than that. Papillary layer. Excellent. Papillary, oops. Papillary. Sublayer of the dermis. Excellent. And as we also talked about, what specific regions have a higher density of them? Excellent. Palms of the hands, soles of the feet, right? The thick skin. Excellent. There you go. Sensory structure 1 on the books. Notice someone else pointed out we also have tactile cells, or remember what are called Merkel cells. What type of information did they give for us? Notice it's got the same name as the tactile corpuscles. Tactile corpuscles, tactile cells also give us that fine touch, right? That discrimination. These are the ones that allow you to pick the quarter out of your pocket. So notice similar names, exact same type of sensory stuff, but are they located in the same place? Where are these tactile cells located?
Is it on the fingertips?
True, absolutely. These are ones that were found in the, again, the fingers, hands, especially the thumb. Let's say that. And where was the other place?
Palms?
In face. But remember, so that was the region or location. But remember, it's also in the specific sublayer. We typically find them in the stratum basal or stratum germinativum. There you go, perfect. Of the epidermis. Excellent. Give me another sensory structure we've talked about. Lamellated corpuscles, excellent. Corpuscles. Excellent, or again, what are also called Pacinian. Right. Excellent. What kind of sensory information did they give us? Pressure, excellent. Right, these are the onion ones that provide that sense of pressure. And where do we find these in the skin? What layer or sublayer of the skin do we find these? Excellent. Dermis is okay, but remember, these are deep, so we want to be a little more specific. It's the reticular layer, sublayer of the dermis. Excellent, 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 excellent. Two more. Conveniently, both of them happen to be on the pretty picture we're looking at right here. What's the next one? Give me another example of a sensory structure we talked about. Excellent. All right, the hair root plexus, which again is ironic. It's called the root plexus, but if you notice from the pretty picture over here, it actually wraps around the bulb of the follicle, but it's still called the hair root plexus, so we'll stick with that. And I'm going to go ahead and cheat and start over again. Hair root plexus, excellent. What type of sensory information does it give us? This should be an easier answer. What does the hair root plexus tell us about? Come on, guys. I know it's been a long 2 weeks, but look at this. After today, we're 25% through the class. We're racing through it. I know this stuff. I'm doing this for you. Got to give me something back. What does the hair root plexus tell you? Sense of touch of what? What do you think the hair root plexus tells you information about?
Hair.
Movement of the hair. There you go. Excellent. And of course, where it's found would be in the reticular layer of the skin, of the dermis, right? And of course, the obvious place is it's around the hair bulb. The last one, remember we talked about, were the free nerve endings. What type of information did our free nerve endings tell us? Excellent. This, remember, was that general sensations, like temperature, like pain, like tickle, like itch, things like those general sensations. And while we talked about them in the papillary layer of the dermis, or sublayer, sorry, sublayer of the dermis, these can also be found in the epidermis. So these free nerve endings can be found in the epidermis as well. And notice this picture has almost all, not all, but it has almost all of these sensory structures on them, right? Notice here are the free nerve endings where our nerve just ends. Notice here is our tactile corpuscle. Here is our Pacinian or lamellated corpuscle. Here is our hair root plexus. The only thing this doesn't show is that Merkel cell. Remember, the Merkel cell or tactile cell would be here in the epidermis, but our illustration doesn't show that. But all the rest of these things can be seen here on our illustration and can also be seen on our models. Again, the only one of these things you're not going to be responsible for histologically, you're not going to be responsible for on a model, is this one, because none of the models, none of the illustrations show them. But all the rest of these you need to be able to recognize on an illustration or a model. All right? So again, despite the stunning silence from most of the class, there's really no new information on here. All of these are things that we already talked about earlier today or yesterday when we were talking about these structures. So there's not new information. We've just synthesized it and put it together here. All right, questions on this? All right, excellent. Let's move forward. Did that. Excellent. The last concept, how are we on time? Oh, we're doing awesome. The last major concept we need to talk about is to talk about tissue repair. Now, today we are focusing primarily on how skin is going to repair, and we will actually talk about those two specific processes by which skin is going to repair, which of course tells you that relays itself to 3 possible essay questions. Describe process 1, describe process 2, and compare the 2 processes. But these repair processes are going to be used not just in skin, but pretty much every tissue we talk about. So we also need to have a basic understanding of the general concept of tissue repair, and that's what we're going to talk about right now. For tissues to repair themselves, basically there are a couple things that are required. The first is we have to remove any injured tissues, any damaged tissues, and replace them. To replace them, remember, as we've talked about, every single cell that is formed in your body has to come from another cell. So we need to make new cells and grow them into that area. And we don't want to do this willy-nilly. So we need chemical signals to control the process. And those chemical signals that we are going to use to control the process are our growth factors and our hormones. Now, there are 2 major types of repair processes. And again, the key to this, we're going to talk about it in skin, but it is important to remember that they are used for all the tissues of the body. All the tissues of the body are going to be repaired by one of these 2 processes. And these 2 processes are regeneration and fibrosis. Now, notice regeneration, the key to regeneration is we are replacing a damaged tissue with the exact same type of tissue. If I'm, riding my bike and I fall down and I scrape up my knee and it's a little bit superficial, it heals. Or if I scrape up my elbow, it heals and it looks the same. The epithelial tissue is there. I damaged that epithelial tissue and I replaced it with the epithelial tissue. And a couple weeks later, it looks brand new, just like it did before. Completely the same. We're replacing damaged tissue with the same type of tissue. However, if a shark takes a huge chunk out of my shoulder, am I necessarily able to regenerate, for instance, all the muscle that is lost there? No. So in that case, we heal that tissue with a process we call fibrosis. Fibrosis is the process where we take a tissue, whatever that tissue is that was damaged, and rather than replacing it with the same type of tissue, instead we replace it with a fibrous connective tissue. And clearly, if I have this highly specialized tissue that is muscle, and I replace this muscle with a fibrous connective tissue, that fibrous connective tissue is not going to look and not going to function the same way that the muscle tissue did. And so as a result of that, we get scar tissue, right? Scar tissue is where the tissue loses its appearance, structure, and its function. Oops. And its function. That's basically what scar tissue is. It is an accumulation of fibrous connective tissue in that area that changes the shape, changes the appearance, changes the function of that tissue. Now, what determines which of these two processes occurs? Every morning, do you flip a coin? Heads, it's going to be regeneration. Tails, it's going to be fibrosis. Do they take turns? Every Monday, it's regeneration. Every Tuesday, it's fibrosis.
Is it
What are the
dependent
two main factors? And that's really the key. There are two main factors that determine which of these two processes are going to occur. What are they? Give me one of them.
Tissue type and severity of energy.
Bingo. There you go. You guys got them both. Absolutely. Well, the first one, as mentioned, is the severity of the injury. As I mentioned, skin, if I just scrape the surface of it, right, is more likely to regenerate. However, if I'm dragged behind a car for 300 yards and massive surfaces of my body are scraped up and abraded, it's much more likely to scar as a result of that, right? If I'm working out and I injure my muscle while working out, that tissue is more likely to regenerate than if I have that shark taking the massive chunk of my arm out. So the severity of the injury is absolutely one of the critical factors. However, as was also mentioned, the tissue type matters hugely as well. Epithelial tissues, as we've talked about, because they're highly mitotic, are much more likely to regenerate. Whereas, as we talked about, cells that don't divide, like nervous tissue and cardiac muscle, they are almost certainly going to heal via fibrosis, producing that fibrous scar tissue. And again, because those are highly specialized tissues, that scar tissue doesn't work the same way as the cardiac muscle, doesn't work the same way as the nervous tissue, and those organs are going to be impaired in their function. Excellent. So which of these two processes is going to occur is going to depend on the severity and the type of tissue. Excellent, excellent, excellent. All right, perfect. Let's talk now about specifically what these processes look like in skin. Starting first with a superficial. Whoops, not wound. With a superficial wound of the epidermis, typically where the dermis is not involved, because remember our epidermis is avascular. There's no bleeding. Now, again, if you scrape up your arm, you may not see bleeding, but you are going to see fluid seeping out. Remember, that fluid that seeps out is that interstitial fluid that we talked about. Remember, that hardened waterproof skin on the surface was keeping it in there. But interstitial fluid will come out of a wound. That seepingness of that interstitial fluid will come out. So it does get wet, but it's not blood. There's no bleeding involved with this process. All right? Now, in this case, as we mentioned, when our epidermis is damaged, the epidermal cells produced a special hormone-like protein. What did we call that hormone-like protein again? I've only been paying attention about an hour and a half ago. What was the hormone-like protein that is produced by the epidermis that is going to help the healing process? Epidermal growth factor, absolutely. So what happens first when the tissue is damaged is that we produce that epidermal growth factor. Epidermal growth factor, as we said, stimulates cell division, but more specifically, it is going to stimulate the stratum basale cells. Remember, those are the ones that are mitotic. Those are the ones that are going to divide more rapidly. So what happens is our epidermal growth factor causes those stratum basale cells to divide. And what happens is when they divide, they don't just divide rapidly. What ends up happening is the cells get really, really large. And as they get really, really large, they then start to migrate across the wound. So instead of growing up like they normally do, now what happens is these cells divide and get very, very big and very, very large, and they start to divide and spread across the surface of the injury. The cells basically get lonely, and so they're looking for friends. And so what happens is this one will divide, oops, wrong button, divide, produce a new cell that grows out this way. This one will divide to produce a new cell that grows out this way, and they will migrate across the surface of the wound because they're lonely and they're looking for a friend. But then something interesting happens. They find a friend. When the cells come in contact with another cell, that changes the growth pattern. What happens is when they make contact with another cell, they stop growing outward. They actually call this contact inhibition. And what happens with that contact inhibition, instead of growing outward, what's going to happen now is now that they've made contact with a friend, they're going to start growing upward. And as they start growing upward, they basically replace the layers like we would expect. As soon as they start dividing up, they form their desmosomes, then they flatten out and they make their proteins, then they undergo apoptosis and die. And at the end of this process, the entire cell layer has grown back via regeneration. Again, the key with this superficial wound, it is regeneration only. And we basically replace the tissue that has been damaged with the identical tissue that was there before. All right. Questions on that one? All right, excellent. Now here's the good news. This process of how the epidermis heals is actually the same for both a superficial and a deep injury. The way the epidermis heals is exactly the same in both instances. So the good news is, if you potentially get the essay question where you have to describe a superficial wound, these are the 4 steps involved in that. If you have to describe a deep wound, when you talk about the epidermis, these are the exact same 4 steps that are going to occur in the epidermis. All right, so the good news is for both processes, you use these same 4 steps because the epidermis heals the same no matter what. What is different about a deep wound is a deep wound involves the dermis. So again, the epidermis is going to heal via regeneration. But now that the dermis is involved, our dermis heals via fibrosis. So with a deep wound, we use both regeneration and fibrosis. And so typically with a deep wound, where the dermis is involved, you often get scarring as a result of that. Now, obviously, one of the big differences between a deep and a superficial wound is with a deep wound, because you're in the dermis, right, there's going to be blood vessels involved, are damaged, and you are going to bleed. You are going to release histamine, and this leads to our inflammatory phase. In the inflammatory phase, histamine is released by the damaged tissues. And as we talked about, that causes blood vessels to dilate, bringing more blood to the area, bringing that redness, bringing that warmth, bringing that heat, bringing that pain to the area. But it also brings the white blood cells so that any type of harmful pathogens that get in here can be attacked and broken down. Of course, as we know, as it bleeds in here, we have those platelets. We have that fibrinogen fibers that are going to come out of solution and form a clot. So ultimately, we need to stop losing blood, and we do that by a blood clot forming. thanks to our platelets, thanks to our other processes of the skin. And once that blood clot forms and we stop losing blood, then we can start the healing process. All right. Questions on step 1? All right, excellent. Let's talk about step 2. Step 2 is called the organization phase. Those fibers inside of that blood clot actually constrict. And as they constrict, they draw water out of it. And so what happens is our clot actually dehydrates. And as it dehydrates, that clot becomes a scab. And that scab becomes a temporary barrier for the healing to occur under. Part of that healing is going to involve new capillaries growing into the area. And those new capillaries basically bring 3 things. They bring new cells, they bring oxygen, right? And when they bring the new cells, they bring the oxygen, and then they bring the nutrients, and or the chemical signals, I guess I should say, to start the healing process. Now, when I say new cells, in particular, there are 2 types of cells that are going to come into this area. The first are macrophages. Our macrophages are our big phagocytes. And remember, phagocytes are the ones that we said are our eating cells, the eater cells. These eater cells are going to do 2 things. Any pathogens that are in the area, they are going to gobble those up and destroy those. But they are also going to remove the dead and damaged cells. So any dead and damaged cells in this area are going to be gobbled up by those macrophages. And what is it that our fibroblasts do?
Creates more fibers.
Makes more cells.
Makes fibers. Remember, fibroblasts makes fibers. They make, and not just any fibers, but in this case, collagen fibers. They are going to start to make lots and lots of collagen fibers. So what's going to start to happen is we're going to start breaking down the dead and damaged cells, and we're going to start to fill this space with collagen fibers. Now, I'm not going to accuse anybody here of picking their scabs, but maybe you've had an injury before with a scab on top and the scab accidentally got scratched off. If that scab accidentally got scratched off, when you look at the skin underneath, does it look like normal skin?
No.
No, it's kind of pink and kind of soft and kind of spongy looking. That newly forming tissue that we see underneath there is actually what is called granulation tissue. So that soft, spongy, delicate pink tissue that's underneath the scab is that granulation tissue that is being formed by those macrophages and the fibroblasts, removing the dead and damaged cells and making lots of new fibers in this area. This is a good thing because it's filling up. Oh, and notice, remember, look at our, let me switch to my highlighter here. Look at our epidermis. Our epidermis is doing its thing. That epidermal growth factor is causing my stratum basal cells to migrate. Notice it's migrating along the surface of the wound, trying to find its buddy so it can come together and regenerate the epidermis. So we see our epidermis doing its thing too at this time.
Question.
Yes.
If you remove a scab like early before it has time to heal, do you cause scarring at that location?
So it can increase the scarring because what can happen is as you remove that scab, that can actually affect the integrity and the wound can actually open up more. And the more it opens up, the more tissue that has to be replaced. So it can increase the scarring, yes. All right. So again, we have that epithelial tissue doing its stuff. We don't want to forget about that. Now, this process continues, and it continues rapidly. And we're making lots of collagen fibers. Again, having lots of collagen fibers isn't necessarily a bad thing in and of itself, but that's not necessarily what was there before. True, this down here was a dense irregular connective tissue, but we may have a different orientation of the fibers that could affect the feel or the appearance of the skin. But remember, there was a lot of other stuff there. There were blood vessels in that area. There were nerves in that area. There were hairs in that area. There were sensory structures in that area, and so on and so forth. And if I'm filling this space with fibers, is there room for this stuff to necessarily come back? No.
No.
So you're not going to get as many big blood vessels in that area, which is one of the reasons scars tend to be a little lighter in color, right? Because there's less blood vessels in that area, so you don't get that hemoglobin in those areas. It may be less sensitive. Glands may not grow back into the area. Hair may not grow back into that area. So it's going to change the appearance of the cell, of the skin, right? Our clot is broken down. Notice our epidermis is now intact and regenerating back. And again, we're making massive amounts of fibers, so much that it can affect the look, the feel, and the function of the skin. All right. Now, eventually, we reach the maturation phase. And when we reach the maturation phase, notice our epidermis is fully regenerated. All the granulation tissue has been removed. And at that point, typically, the scab sloughs off. Then typically what happens is that our fibers, all these collagen fibers contract, forming this tightly packed structure, and this tightly packed structure can become that scar. Now notice, and again, you haven't necessarily thought of it in these terms, but you know this. Notice we started with this massive wound. Let's go back all the way to the picture. And we just allowed it to heal. If you have a big, huge gaping hole like this, and you just let it heal, then yeah, you're going to have a massive amount of scar tissue in there. So what does the doctor typically do when you have a big, massive wound like that?
Sews it back together.
Yeah, they put in sutures to sew it together. So instead of having to fill this much space with new scar tissue, by bringing the tissue together, we can minimize how much scar tissue is going to form, and we can dramatically decrease the amount of scarring that is going to form. All right. Questions on that? Excellent. Just that. Go ahead.
So what is pus to this process?
So pus occurs when there is an infection. If an infection occurs, what happens is you get an increase in the number of white blood cells that come to the area. And as those white blood cells are fighting that infection, that causes pus formation to occur. Now, again, pus is not the same thing as that seeping that we were talking about, right? Is like I said, if you abrade your arm, if you scratch up your arm, you can see some seeping of it. But as we talked about, that seeping is typically clear in color. So it's more of a clear fluid that is coming out of it. That is typically interstitial fluid, whereas pus is typically more yellowish or whitish in color, often can have a smell associated with it as well. And that's because, again, your white blood cells are fighting that infection. Great question. Any others?
What? Never mind.
All right. Well, then in that case, that is everything we need to do from a lecture standpoint. Now, as I mentioned, I want to make sure we emphasize these things and put these things together. So the last part of this that I want to make sure we emphasize, let me do this, is to come back to our histology. Actually, I'm going to cheat and do two more things. This and this and this to, wait, no, hold on. Copy. Excellent. One of the things I was hoping someone would mention yesterday when we were going through this, but didn't, was that as you looked at your handouts, one of the things that you were responsible for on your epithelial parts is where it talked about the membranes, right? But notice we didn't mention that when we were talking about it. So let's go back one last time with a little bit of time we have left to our histology slides, to something we were talking about before. When we looked at this before, and where's my highlighter? Identify the tissue type again.
Stratified columnar.
Is it stratified columnar?
Just kidding.
Simple columnar.
Stratified.
Simple
Simple.
columnar. This is a simple columnar epithelial tissue, right? So what if instead I took my pointer off of this slide and I asked you to identify the membrane? What membrane is this?
I can't see the pointer.
Exactly. There isn't a pointer on here. What you see in the field of view in front of you is a membrane. What membrane is this? I guarantee you this is a question that is on your, not necessarily this slide, but I guarantee this is a question that is in your bank of the lab exam. Absolutely. This is a mucous membrane. How do you know that?
That goblet cell.
Okay, absolutely. The goblet cell is a dead giveaway that this is a mucous membrane. But we can also take it one step further, right? What type of tissue is this again?
Column or
Non-characterized.
No, simple columnar.
Simple columnar.
And where are the places where we find simple columnar again? One location where you find this tissue.
Stomach.
Stomach, small intestine, large intestine, right? Distal part of the esophagus. All of those are part of the digestive pathway, right? Our digestive system. So its lumen is open to the outside world. And if its lumen's open to the outside world, what type of membrane is this? Mucous membrane. Notice that means there's a couple other questions I could ask here as well. Identify this tissue type. Areolar, excellent. Because we know all mucous membranes have areolar connective tissue as the part of it. Remember, a membrane is made up of an epithelial tissue and a connective tissue. Our epithelial tissue is simple columnar. Our connective tissue is areolar. And with that exact same pointer, I can also ask the question, identify this tissue based on its location. Lamina propria. Perfect. Identify the membrane. Identify the membrane. Excellent, perfect. This is indeed a mucous membrane. Excellent, right? How do you know? I don't see a goblet cell in this one. Exactly. This is a ciliated pseudostratified columnar epithelial tissue. Where are the locations we find that again? Nose or trachea. Those are open to the outside world. So this absolutely is a mucous membrane. So identify this tissue based on its location. Lamina propria, excellent. Identify that tissue type.
ciliated tissue.
No, this one down here, sorry. You might not be able to see my pointer. This one down here. This is the ciliated pseudostratified columnar. But this down here, let's do this to make it easier. I will make a nice big blue arrow. Okay, no, that's not big, but that works. Identify this tissue based on its location.
The transitional epithelium.
Nope. What is this tissue based on its location?
Auroral tissue? Aero
It is an areolar connective tissue, but notice the question I asked. I didn't ask you to identify the tissue type. I asked you to identify the tissue based on its location. You are correct. This is an areolar connective tissue, but its location is part of a mucous membrane. And as a part of a mucous membrane, what do we call an areolar connective tissue that is part of a mucous membrane? Yep, Alexa's getting it, the lamina propria. Conversely, I could ask you to identify the tissue type, and then your answer would be?
You've lost us.
Apparently. The arrow. Like I said, I guarantee questions like this, these are exactly what you're going to see on the lab exam. You're going to see a slide that looks just like this with an arrow just like this, and it's going to ask you to identify this tissue type. What tissue type is that? Areolar. Excellent. I encourage all of you to cheat off Alexa during the exam. They seem to have this down. Excellent. The rest of you got some studying to do this weekend. And if you haven't figured it out, I am beating a dead horse. So let's look at a third example. I'm going to move my arrow out of the way so you cannot see it. Identify the membrane. What membrane is this? Mucous membrane. Excellent. How do you know that? How do you know this is a mucous membrane? True. Well, but remember, all epithelial tissues are open, but more specifically, what tissue type is this? Excellent.
Transitional epithelial.
Remember, look, flat cells with nuclei on top. It is a non-keratinized stratified squamous epithelial tissue. And as someone mentioned, it's open to the outside world. Remember, this is found in the oral cavity, in the esophagus, in the vaginal canal, in the anus. These are all things that are open to the outside world. So that makes this a mucous membrane. And that makes this tissue down here. What tissue type is this tissue down here?
Areolar.
Areolar. Excellent. And identify the tissue based on its location. Lamina propria. Perfect. Some of you have got some studying to do this weekend. Again, half of this exam, if you think of the amount of time we spent lecturing, half of this exam is tissues, is membranes. I warned you, this is the hardest part. This is the part that is least intuitive. But you have that study guide to help you. We're going through this together. Make sure you've mastered this by Monday.
Question.
Yes.
If it's non-keratinized on top, does that automatically make it a mucous membrane?
Absolutely.
Okay.
Notice all three of these, and I'm going to go ahead and move this arrow again. Notice all three of these slides. This one with the non-keratinized tells you right away it's a mucous membrane. This one with the ciliated pseudostratified columnar tells you right away it's a mucous membrane. Even this one with a simple columnar. If we think about the location where these tissues are located, all of these tissues line organs that are open to the outside world. So based on their epithelial tissues, we can tell all of these are mucous membranes. And once we know they're mucous membranes, we know everything else about them. We know that they're on top of areolar connective tissues, and we call that areolar connective tissue a lamina propria based on its location. All
Question.
right? Yes.
If they're mucous membrane, does that again automatically make it a areolar connective tissue?
Yes, because remember we said regardless of what epithelial tissue is on top, all of our mucous membranes have areolar as their connective tissue. And
okay, and do we, when we mentioned it, what was the difference between the areola and the lamina, you said?
one is the tissue type, one is the name we give its tissue type based on its location.
Oh, got it.
What I encourage you to do if you're having trouble with this is go to the membranes part of your textbook and read that, and go back and watch the lectures again. Again, remember with these lectures, you don't have to watch all 4 hours of them. You can just fast forward to the part that you need to focus on. I would encourage you to go back to those membranes again if you're struggling with these things. All right, I don't want to spend any more time on these membranes because I want to talk more about what we did today, and that is our skin, right? Because again, a lot of questions that could be asked here. So let's take a look at this and start asking some of these questions. We only have a few minutes left of class, so let's rapid fire this, and you guys got to give me the answers. You stop answering, I stop asking the questions, and we end the lecture, okay? Identify the type of skin. What type of skin is this? Can't answer the easy questions. You guys are in trouble. Thin, there you go, excellent, thin skin. Excellent. One region of the body where you would find this skin.
Forearm?
True, although we don't want to use
Face.
we don't want to use things like arm and face. We want or chest. We want to
Eyelid.
use appropriate anatomical terms. So instead of forearm, what could we say instead? Brachial region or antebrachial region, right? Clavicle region, I'd accept that. Absolutely. All of those are good. Thoracic region, buccal region, excellent. All of that is perfect. All right. Here, I'll use this. Identify this general layer. Epidermis, perfect. Identify this specific sublayer. Stratum basale, excellent. This specific sublayer.
Stratum spinosum.
Spinosum, excellent. This specific sublayer. Stratum granulosum, perfect. And this specific sublayer.
Stratum corneum.
Excellent. All right. Notice we didn't also ask the easy question. I asked you to identify this general layer, but what if instead I asked you to identify the tissue type? What tissue type is that? Identify the tissue type. There you go. Keratinized, squamous, stratified, perfect. Epithelial tissue, want to throw that on there as well. Excellent. Let's do some more. Identify the cell.
Keratinocyte.
Excellent. And this one? You can say it again.
Oh,
Keratinocyte.
keratinocyte.
Excellent. And this one?
Keratinocyte.
And this one?
Dead keratinocyte.
True. And dead keratin is fine, but keratinocyte is fine as well. And what about this one?
Melanocyte.
Melanocyte. Notice here, and let's clear that and actually draw a little circle. Notice there's a couple of them here. There's another one here, another one here. Notice this one has a lot of melanin in it, so we can see that dark pigment. Remember, we talked about how this is dark pigmented skin. So notice this happens to be an area where that has a lot of those melanocytes. And again, remember, it's that clear cytoplasm that is going to allow us to be able to distinguish and recognize them. All right, a few more quick questions. Identify, where's my highlighter? This structure. Papillae, excellent. Notice I can leave that green box there and I can ask you 3 other questions. Actually, 4 other questions. All right, so 4 questions. Okay, structure with papillae. What general layer of the skin is this? Dermis. What specific layer, specific sublayer of the skin is this? Papillary sublayer. What tissue type is this? Excellent. Areolar. Perfect. I could grab my highlighter and move it down here instead, and I can at least ask 3 of those questions again. What general layer of the skin is this? General layer. Dermis. Specific layer or sublayer, more specific sublayer, reticular. Patricia absolutely has that right. Tissue type. What tissue type is this? Dense irregular connective tissue. And again, I'm fine with you guys abbreviating it now to be quick when you're typing, but remember on the actual exam, write it out. Do not use DICT or KSSET on the exam. Make sure you're writing these things out. Beautiful. And notice we could ask all of those questions again right here. Type of skin.
Thick skin.
Thick skin. Excellent. General layer of the skin.
Epidermis.
Excellent. Epidermis. Tissue type.
Keratinized.
Keratinized. Finish it off.
Epithelial.
Keratinized stratified squamous epithelial. Remember, you have to have all of that. Keratinized stratified squamous epithelial tissue. If you just write keratinized, you only get partial credit for that. So make sure you have it all. All right. Sublayer. Oh, here, the red light just doesn't show up good on that. So let's switch back to this. Sublayer. Stratum basal. Excellent. Sublayer. This is the exact same questions I just finished asking. Spinosum. Excellent. Sublayer. Granulosum. Excellent. Sublayer. Whoops, hold on. Let me redraw that one. I didn't like that. Sublayer. Stratum lucidum. Again, it's good to say stratum on those as well. Sublayer.
Stratum corneum.
Stratum corneum. Perfect. Excellent. Structure.
Papilla.
Papillae. Excellent. General layer. Oh, I like that. Dermal papillae works. And that gives us the answer to the next one. General layer is dermis. What sublayer is this? Papillary sublayer. Excellent. Tissue type?
Dense irregular.
Now remember, the dense irregular is going to be lower than that. This is the areolar. Remember, the papillary is made of areolar. Notice this particular view, because we've got all the epidermis, we don't really see the reticular layer, maybe the teeniest bit of the beginning of it here, but we're really not seeing the reticular layer here, not seeing the dense irregular here. All right? So there you go. Notice those are all questions I guarantee are in your collection of databases that you're going to be looking on. And because we did it today, let's do the same thing for glands. Notice I could show you this gland, and there are many questions I could ask. Let's start simple. Identify the general class of gland. I know we just did this today, but like I said, the test is on Monday. So work with me. How are we on time? We're okay. What is the general class of gland that you're looking at here?
Eccrine. No, that's specific.
Eccrine would be specific. You're absolutely correct. This is an eccrine sweat gland. But the general class, excellent, Jessica's got it, is sudoriferous. Excellent. So sudoriferous is the general, but you are correct. If I asked you for the specific gland, you would say eccrine sweat gland. Remember, I could ask you for the structural classification. What is the structural classification of this gland?
Simple coiled tubular.
Perfect. What is the functional classification?
Merocrine.
Merocrine. Excellent. Perfect. right? Merocrine is the mode of secretion. Excellent, perfect. Substance it produces?
Watery sweat.
Watery sweat. Region of the body where you would find it?
Your axillary region.
Axillary region. And again, remember, is there a wrong answer to that question?
No.
No, because everywhere. Now again, if I ask you for one region, do you want to write everywhere as your answer? No, just give me one. But the good news is, as long as you give me one, you're not going to be wrong. All right? Compare this to this. Notice some of the answers to this question are going to be the same. Identify the general type of gland.
Pseudoriferous.
Pseudoriferous. Identify the structural classification.
Apocrine.
No. Structural classification.
Orbital.
What is the structural classification of this gland? There you go, simple coiled tube. What is the functional classification or mode of secretion of this gland?
Merocrine.
Merocrine.
Okay.
Notice those three answers are the same for both of these. They're both generally pseudoriferous. They're both simple coiled tubes. They're both use the merocrine mode of secretion. But as many of you have already pointed out, you've seen that big, large lumen, so you know the specific type of gland is the apocrine gland. And what does that apocrine gland produce?
Watery sweat.
Not the watery sweat. Which one does this produce?
Sebum. Sorry.
Not sebum either.
Organic sweat.
Organic sweat. This is that organic sweat. Excellent. And one region of the body where you'd find it? Axillary, right? Anogenital, buccal, thoracic, right? All of those are examples of regions where you'd find it. Because remember, this is the one that is associated with our secondary hairs, those hairs that show up at puberty. Yeah, so much good stuff here. Excellent. Let's stick with the glands for right now. Identify this specific type of gland.
Ceruminous.
Now remember, the ceruminous and the mammillary, I'm not going to hold you responsible for histologically. So you need to know ceruminous glands are found in your ear holes. You need to know mammary glands are found in the breasts, but you're not going to need to know them on a microscope. What is this specific type of gland? Sebaceous. Excellent, right? Notice right here are two hair follicles. This one actually even has a hair in it still. So notice the fact that these are associated with hair follicles is a dead giveaway that these are sebaceous glands, and they're these big clear cells with the big prominent nucleus in the middle, because these sebaceous glands produce what substance? Sebum. Sebum, excellent. Perfect. Structural classification of this gland. Branch to center or a velar. Excellent, perfect. Mode of secretion or functional classification.
Holocrine.
Holocrine, excellent, where it explodes. Perfect, excellent. Function of the sebum. What does sebum do for us?
Moisturizers.
It moisturizes, waterproof.
Maintains the integrity of the hair and the skin. Perfect. Excellent. So we have all of that. And notice also, as I said, we have our hair and our hair follicles here. These are things we, as I mentioned, you need to know histologically as well. Now, let's start this one because this one's easier because this one doesn't have the hair in it. So it's just the follicle. And remember, as we talked about, 3.5 layers to the follicle. This one here is our internal epithelial root sheath, our external epithelial root sheath, and then right here at the line between them would be where the glassy membrane is located. This particular microscopy slide doesn't show the glassy membrane, but often it will be here. This particular one doesn't show it, but this is where it would be between the epithelial tissues and these fibers. Notice how, again, there's a lot of fibers in our dermis, but these fibers are wrapped around the follicle. These fibers are wrapped around the follicle. So this is our dermal or connective tissue root sheath. This, of course, is the hair. We can't see the layers of the hair the same way, but we know the very center is the medulla. This is the cortex, and then that dark line on the outer surface would be the cuticle. So while we can't histologically really differentiate the hair, the follicle we definitely can tell histologically. All right, questions on that? All right, 2 more fun things. What am I looking at here? Any idea? What do you guys think this is?
Almost looks like the heart.
Kind of, does kind of have a heart shape to it. Close, yeah. Or more specifically, this is the hair bulb. This is the bulb of the hair. You are right, these cells located right here would be the matrix. And notice here we have the dermal papillae that brings the capillary, brings the blood vessels to it. Notice here we see the medulla, the inner part of the hair. Here we see the cortex, the outer part of the hair. And these little cells out here is where the cuticle is starting to form. Remember, the cuticle doesn't form till much later. So it's starting to form. It's not complete yet. We have a little bit of the incomplete cuticle. And then our cortex and our medulla and our papillae. This is the bulb of a hair. Absolutely. Excellent. And one more. What do you think this is?
Bone.
Not a bad guess. Of course, bone's not one of the things you're responsible for in the skin. So while it kind of has a bone-like, kind of looks a little bit like that osteon of the bone, what we have is an onion-like structure with many, many layers to it. Didn't we say something was kind of onion-shaped with many, many layers?
Nail?
Close.
Fingerprints. I mean,
Fingerprints is a good guess as well, but no, actually, this is a sensory structure. This is the lamellated or Pacinian corpuscle. Remember, I said it was made up of many, many layers of plasma membrane. And when those plasma membrane layers are squeezed or stretched, that sends an electrical signal to your brain and you feel pressure. So I just wanted you to see that these Pacinian corpuscles or lamellated corpuscles really are made up of many, many layers of membranes forming that pressure-sensitive sensory structure. So this is that Pacinian or lamellated corpuscle. All right, excellent. With that, that is everything you are responsible for, lecture-wise, lab-wise, everything you need to know for the exam. And we still have a good 15 minutes left. Excellent. Any questions on any of that? Again, I know I went through it fast, but remember this is recorded. You can go back, but I also want you to go back to the lectures and look at these theories. If you think about it, a lot of the things we've done for the past 2 hours has really just been synthesizing the information, condensing the information, expanding on it a little bit, but really taking concepts we've talked about and putting them together. And that's what you're going to need to do on the exam. And again, that's the challenge with a timed exam. The more familiar you are with this material, the easier it is to put these pieces of the puzzle together. So you really need to spend a lot of time between now and Monday familiarizing yourself with this so that you don't have to stare at that histology slide for 10 minutes just to figure out what the heck it is. You're going to recognize it right away, and as soon as you recognize it right away, then you can actually focus on answering the question. Great question. No, because we've run out of times, both burns and cancers will not be a part of your exam. Normally we get a chance to cover it and we talk about those burns, we talk about the cancers, we talk about the rules of 9. But we didn't do that in any of our labs, and we ran out of time because summer. So no, those will not be a part of this exam. So you are not responsible for those on the exam. All right. Great question. Any others? All right. I am around this weekend. Obviously, Sunday is Father's Day. So I'm sure my children will dote on me for at least 15 or 20 minutes. But other than that, I might have a little bit of free time. I'll definitely be available tomorrow. I'll be less available on Sunday, but I know we have a Monday test, so I will be available. So if you have questions or anything like that, feel free to email me and answer those. Otherwise, I wish you the best of luck on Monday. And remember, Tuesday we're forming groups for group presentations, so make sure you're there for that as well. Otherwise, study hard, good luck, be safe, take care of yourselves, and Monday, good luck on your exams. All right, guys.
All right, thank you.
Thank you.
Thank you.